Yeah, let's get started. It's 9:04 already. So good morning, everyone. How is everybody doing today? Welcome to our Utah Onion Association meeting, a little bit different this year, as we are having our Northern Utah Commercial Vegetable Grower meeting. So we would like to thank you guys for being here today. Thank you for all the speakers. Thank you for the effort, in being here. Thank you for those who are on Zoom. So we are actually recording if somebody missed the meeting so they can watch it later. The link will be posted on the Vegetable Production Lab. So new people and then new tasks, right? So we are gonna have a new website where I can, post everything there. So keep you guys posted on the new events. So anyhow, a little bit about myself. My name is Milena Oliveira. I am Brazilian, so I'm the new Extension Vegetable Specialist from Utah State University. Great pleasure to meet you all. For those who I did not have the chance to meet, last year while I was going around the country trying to meet as many growers I could. I am, as I said, Brazilian, so you judge by my accent. I grew up in a small farm in the northeast area of the state of Brazil. So it's pretty much dry. My whole career is based on dryland agriculture, so I know what you guys face here. What else? I have master in plant physiology, bachelor's and PhD in agronomy horticulture. I have experience not only on vegetables, but also on fruits, ornamentals, row crops, dryland cropping systems, and so on and so forth. I spent some time before coming here, between my PhD and here, I spent some time in Israel, where I was studying agriculture and biotechnology for drylands. So trying to, increase the crop performance, crop resilience, trying to identify or develop sustainable strategies to, dryland agriculture. Anyhow, what else? Before coming to Utah, I was in Western Nebraska doing a postdoc in dryland cropping systems, working on the long-term impact of fallow replacement, on the subsequent winter wheat yield. So I have experience on, row covers, again, cover crops, crop rotations, and so on and so forth. I think that's it. The main purpose of this meeting is actually to not only bring knowledge to the onion growers, but also to the other vegetable growers. But this meeting is actually not limited only to the big growers, to the, the commercial ones. So small acreages, gardeners, small, beginners are more than welcome to come. We actually don't know exactly how we are gonna do, because this year is the first one. So you guys will tell us how are we gonna do that in the next few years. If you wanna have it every year, if you wanna have it every 2 years, online, in the middle of the summer. So you guys decide, and please let us know what do you want us as Extension, to do for you guys. We are here to serve. And I especially really like to meet growers, learn, because we learn in a daily basis with you all, especially with your challenges. So I think that's it for now. We have 3 little small things to say before we start. So we have a list of information. So we ask you guys to please update your phone number and email. Some growers that I know, they reach out saying that they did not get the email. So probably they have another email. So please update the list. What else? We have a survey, which will be part of my formal needs assessment. So I ask you guys, please, to fill out. The forms are over there. So you can use the break, to do so. Otherwise, I can send you online too. We have 7 speakers, including myself today. I would like to thank you all for, being willing to give these talks in a very short period of time while we organize this meeting. Most of the topics you will see here today they were actually a result of our survey that we sent last year, and also some suggestions that, some growers reach out that they needed some information on, okay? So welcome again, and let's start with our first speaker. So Corey Ransom is our weed specialist from the Plant Soils and Climate, my colleague. Thank you, Corey, for being here. I think I will pass the mic.
She showed me where the off is so that I can say things without being on mic. I appreciate the opportunity to come visit with you this morning just a little bit, and where this is kind of a new meeting, a lot of my comments are a lot more general and hopefully give you some resources. But if you have more specific questions, I'm always happy to help in areas that you might need specific help with weed management. Just keep in mind that in my role here, I spent my first 8.5 years with Oregon State University in the Treasure Valley, got a lot of experience with specialty crops there. And then coming here was drawn into everything else, right? So I work everywhere from agency level weed management down to, homeowner kind of weed management issues. And so unfortunately, I don't get to spend as much time as I'd like in kind of the vegetable production area. Just a personal note, I want to point out, I'm really pleased to be able to present here today because my dad was actually discharged from Bushnell Medical Hospital, which sat on this site after he was injured in World War II, fighting outside of Metz, France. He actually lost his leg and as a result of that, and came here. Bushnell Medical Hospital was a rehabilitation hospital for amputees, which worked to provide them some life skills. I'm grateful my dad survived because he turned out to be a wonderful dad and a great man. Okay, today I'm going to kind of talk about some general concepts of integrated weed management, talk about what I see some of the challenges that we face going forward, talk a little bit about some of the weed management resources and opportunities that are out there, and then just touch on some changes happening to specifically Roundup for homeowners, just in case we have hobbyists here. And if we think about, again, kind of the pillars of our weed management approach, we know that we have cultural, biological, mechanical, and chemical options or things we can bring to bear. And when we think about biological, a lot of times that kind of goes away, right? We don't really have animals we can graze in our vegetable production systems. We can't wait for insects to come and feed on invasive species. So that leg of the stool, if you will, is kind of taken away. And when we talk about cultural, sometimes when we're working with a crop like onions, crop competition is a cultural management strategy. Well, if your crop is not competitive, then you don't have that advantage. So then you end up having to lean on some other things. The other way we kind of talk about weed management are indirect and direct weed management. And indirect are really things that we do in our crop management process that can impact the weeds. So it's not specifically done for weeds, but it can have a really great impact on weeds. So if you just think about, drip irrigation, where you're applying the water directly where the crop is planted, less water available to weeds. So that in an indirect way can have a big impact on weed management. And then of course we have all of those direct weed management tools that we use. And so just thinking about, things like prevention. So making sure that we're not bringing new weed problems into our systems. It's amazing how often a new weed species shows up as a result of compost that's maybe improperly composted, or mulch, or other materials that we bring onto our farms or our land. I really loved when I worked in the Treasure Valley that they had these screens on their irrigation systems that as they bubbled up, the water would bubble up, flow on these screens. And it was amazing how many weed seeds came out of those screens before they ever made it to the field. So some important things just to think about that prevention. A lot of times we save ourselves a lot of problems by working in a preventative way. Just talking about cultural weed management, it's not a new concept. A German researcher in 1939 talked about the fact that cultural weed control should form the basis of all of our weed management, and everything else is kind of secondary to that. Unfortunately, again, we've really become dependent on herbicides because they're so dang effective. But as I talk about one of the challenges later, we're starting to get resistant weed populations. And sometimes the only solutions to those are to go back to the basics of trying to use other things other than chemical weed control. Just some thoughts here. Again, cultural things we could do, growing competitive crops, right? So picking varieties or crops that can compete with the weeds that we face, targeted irrigation, putting fertilizer directly where our crops can utilize it and the weeds can't. Again, a picture back from the Treasure Valley. I love this. You can see where they've bedded up the onion field in the fall. So then in the spring, they can come in and spray out weeds that have germinated. They don't have to wait for to be dry enough to make those beds. As soon as they can get across the field, they can get that planted. So cultural, time of planting, right? Stale seed bed approach. So this idea that you can kind of clean sweep without large disturbance and not bringing new weed seeds to the surface. So really important. And this really plays in then to this other idea of planting dates and things we can do before weeds germinate. So just being aware of what is my most problematic weed, and maybe if I delay 2 weeks, could I clean the field up with herbicide or with flaming or something before I plant and have less of that weed as a problem. So a lot of research. What's hard with this is these kinds of research are always done in specific areas. So you as growers and producers would have to know when particular weeds germinate under your particular circumstances. Lots of innovative approaches. I've stolen some of these, but lots of tools that we can use. I really love this one where you're laying face down to do your hand weeding. And I actually have a picture also. It's from the Dutch Institute of Agriculture that shows a bunch of people side by side on a big frame, all weeding their own row. And I said, instead of speed dating, it's weed dating, right? Just switch positions as you move down the row. My students who did an onion project years ago had so much bindweed in the plots, they spent basically the entire summer trying to hand weed it back. And they took some old sprayers we had. We hung weightlifting benches on ropes on them, and they would roll the tires along. It was a low-tech version of this, so that they didn't have to be crouched all day long. Things like mulches can be highly effective. Sometimes you just got to get the hoe out. So I love this cartoon. Every person in this world who has ever amounted to something started by using a hoe. And so Linus says, now I'm on the road to greatness. And I didn't start out with a hoe. I started out with a shovel cutting thistle in my dad's pasture. So that inspired me on to something better. And then herbicides can be really critical. So just an onion field with no herbicide, different herbicides and different timings. So it's not just the fact that we have herbicides, but how we use them, the combinations we use, the way that we utilize them in order to make sure that we have effective weed management. Let's talk about some of the challenges. I think some of the challenges are that we just continue to have new weeds and their ability to move into our areas grows. Just talking about how I spent a lot of my career in the Treasure Valley working on yellow nutsedge. And when I came here, it was like yellow nutsedge used to be a problem, but as farms moved out of the Kaysville area north, they kind of moved away from that. Well, I hear maybe, there are some issues coming in. So we can get away from them for a little while, but eventually they catch up. The rapid spread of herbicide-resistant weed populations, I'm going to focus on that. Increases in regulation and disapproval of herbicides and GMO technology in general. And that's corresponding with kind of a slowdown, right? There's not a lot of incentive for companies to put millions of dollars into trying to develop new herbicides because it just doesn't pay back for them. And then one of the biggest challenges, which is ongoing, is that the challenges you all face with commodity prices, input costs, and things like that as you try to make those decisions. So this chart is from one of my colleagues, Ian Heap, and he kind of tracks herbicide resistance, but this one is specifically showing discovery of unique modes of action. And what this shows, which is really kind of disturbing, is that since 1980, there's only been one unique mode of action discovered. Okay? When we look at resistance, keep that 1980 in mind when you look at the rate of herbicide resistance. Here's that. So total number of unique cases of herbicide resistance, pretty slow in 1980, but since then, just an exponential increase in the number of species that are resistant to these herbicide products that we have. And we're not replacing this, right? There used to be a thought process, well, the industry will develop new herbicides that will take care of this resistance that's developing, but that is not actually happening. In Utah, we've done some surveys and showed that we have widespread resistance to the ALS and herbicides, which include things like Pursuit and Raptor in alfalfa, and things like Telar and Escort and Range, and those same molecules that are used in kind of our grain crops. More recently, we discovered some kochia that was no longer responding to glyphosate or Roundup. We've done those tests, and we've documented that resistance. I'll show you a couple slides. Just this past fall, we were called out to a dryland farm to look at some downy brome or cheatgrass, which appears to no longer be responding to Roundup. And so the bright side of that is Eric Westra, a new weed scientist in our program, has spent a lot of his career working on herbicide resistance. So we're expanding our efforts to document resistance and then hopefully develop strategies as those weeds become resistant that will help us manage them. I'll just mention here, back to integrated, right? I've never seen a herbicide resistant weed that was resistant to a shovel or a hoe, okay? So when you see that one escape, it's probably worth going out and taking it out, not taking a chance that it may have actually survived a herbicide application and potentially could cause problems well into the future. This is just a dose response in the greenhouse. So the top is a known susceptible population. And amazingly, kochia in the greenhouse is harder to kill than it is in the field. Some plants it's the opposite, but in the greenhouse, kochia actually tolerates higher rates. But you can see we get complete death at about that 33 ounce rate of the susceptible population. Are just to decrease biomass by half, the resistant populations, they're up around 100 fluid ounces, and that's for 50% reduction in biomass. So you can see this is not a problem. You just up the rate of Roundup, right? You can't kill it with Roundup at labeled rates. So how can you manage this? So I'm just going to jump in here. Again, it's all these things we talked about, these concepts of utilizing tillage, utilizing competitive crops, making sure you're not introducing things. And then when we do use herbicides, it's the idea that we should rotate different modes of action between years, or maybe actually use multiple modes of action in a year. That is easy to talk about if you're growing corn, where you have a plethora of herbicides and modes of action. That becomes really difficult in some of our specialty crops where we have one or two key herbicides that we rely on. So that's maybe where crop rotation becomes the opportunity to work in that. And then just finally back to this idea, and we're going to have to update some of the information I'm going to show you from the USU Extension website. In August, EPA basically announced an emergency suspension of all uses of Dacthal or DCPA, which if you look at most of the uses of Dacthal, it's almost all in specialty crops, right? No big, large crops that it's really ever been registered on. This suspension prohibits any further use of the product and actually has some rules about even transportation unless it's being transported back to the manufacturer for return. So if you have Dacthal, you shouldn't use it anymore. And if it was recently purchased, you might be able to get some money back from AMVAC for that. So that's really dark and doomsday. Let's talk a little bit about some of the resources that we have. Again, I think we see that technology is making its way into agriculture, right? So we have some good advances that are allowing us to be more site-specific in our application. We have even nozzles that spray when they sense a weed. So in bare lots, you know, as they drive that boom across, it only triggers the nozzle if there's green material there to be controlled. We have things like drones and lasers and other approaches that hopefully can kind of step in and take the place of that reliance that we've had on herbicides in the past. As far as herbicide development and use expansion, you know, it's really dealing with what we currently have. There are two companies who have announced new modes of action that they've discovered. And unfortunately, one of those is for grass control in rice. So I'm not sure how that one's going to, you know, benefit us. I'm not aware of what the other one's use patterns might be. But again, then some ideas of how we can use existing products with maybe herbicide resistance traits. That's been really hard, again, to bring into vegetables, right? Things that people eat, they're a little less open to some of those GMO or even non-GMO traits. So another resource I want to point out to you is this new chart made by a committee called the Take Action Committee. And it shows modes of action and trade names and even some premixes. Again, this is built in the Midwest. We really hope that we're going to modify this so that it's available for growers more in Utah. And it's going to have more soybean herbicides on here than it has specialty crop herbicides. But just to give you a good idea of that, if you're interested, you can either search Take Action Committee mode of action chart or this QR code. We'll take you to that chart, and it shows you how the different herbicides work, and more importantly, gives you a site of action, what we call a group number. And so that's that number on the left-hand side, which is a group 1, a group 2, a group 9, which identifies those herbicides. And those group numbers are on most of our herbicide products now that we get commercially. So you can see it's a jug of Roundup. I don't need to know its enzyme that it targets. But if it says group 9, I know that any how can I maybe mix up some of the herbicides that I'm using? Another resource that I think can be valuable is that I'm the liaison for the state of Utah to the Western IR4 program. And what IR4 does is it develops the tolerance or residue data necessary for a chemical company to expand its label onto small acreage crops. So if you're a big company, it makes a lot of economic sense to register a product in corn, where you're going to sell that product on millions of acres. Makes a lot less sense to spend all the regulatory monies to get a really small acreage crop. And that's where IR4 with federal money steps in and helps provide that data. And so if there are specific needs and potential tools that could help us address those needs, we can go onto that site, put in a request. That request gets considered. If it's important enough, it gets ranked, it gets funded. They generate the residue data that goes to EPA, and then the company can add that crop or crop group to their label. So pretty great program. I don't know if it will survive, but it has, if you look at a lot of the products that we have in our specialty crops, a lot of that data has come through this program. It hasn't mostly come from big corporations. You got a copy of this, so I won't spend much time on this, but the Utah Vegetable Production Guide at Utah State, a good resource. I'll just warn you, I probably didn't give any kind of review of that. Like when you go to onions, there's some key herbicides that aren't listed there, so it's not fully comprehensive, but it's a really great resource and has a lot of great information, not just about pest management, but about the growing of the crop. And so I'll let you find that one. Here's another one that I've used a lot throughout my career. It's the Pacific Northwest Weed Control Handbook. It also has really specific pest management suggestions on there, some general principles again. And I'll just warn you on that one, it's written for the PNW. The PNW has some pesticide labels that are specific to them and do not include us here in Utah. So if you look in there and say, oh, here's a herbicide I could use for weed control in onions, you need to go to that label and make sure that label says that you as a grower in the state of Utah can use that herbicide, okay? And there's, if you want to link to that, there's a QR code that will take you to their general page. And again, a variety of specialty crops. I also use this a lot for agronomic crops and other sites. So, application technology. So how frustrating it is to need to get in to do a spray, but the field's too wet, and maybe to be in an area that has houses a little too close. Well, we've seen a lot of companies pop up that are now making pesticide and even fertilizer applications with drones. And pretty exciting that maybe there's a solution to some of these fixes that you find yourself in. And I don't know, how many of you had a drone application on your farm to this point? Yeah, pretty amazing, right? The technology and kind of the smaller scale. And I think specialty crops is a really great niche where maybe that technology helps a little bit. Okay, so last is kind of a warning for anybody who might be a homeowner and is buying their Roundup specifically from a box store. These two jugs look alike, but they are not the same. So as a result of the lawsuit where Bayer and now, Monsanto and now Bayer have paid out billions of dollars in what some people might consider frivolous lawsuits. I'm not going to put my 2 cents in that hat. I guess I already did. But so as a result, most of those suits are coming from homeowners, right? And so Roundup in the box store will no longer contain glyphosate. So when you buy a jug of Roundup from that box store, it's not going to have the properties of glyphosate. And what they've replaced glyphosate with, so the top ones are all of the glyphosate ones, okay? And if you look down, the new ones, in order to get the same level of control, are going to contain triclopyr, fluazepop, and diquat. So if you're used to spraying your garden and then planting immediately your tomatoes into your garden, guess what? Glyphosate was perfectly safe to almost immediately replant. You're not going to find anywhere where you can replant vegetables, or it's suggested that you even use that in that garden setting on this new label. And keep in mind that commercial product, that probably doesn't apply to home use, right? You probably can't find on that label for use in your home garden. So the commercial grower will still have access to glyphosate and Roundup. Just keep in mind, read the label. Roundup is now a branding and not a product, right? We have Roundup for lawns. What does that mean? It also has never had glyphosate in it, right? Because glyphosate would kill lawns. It has broadleaf materials. So read those labels, don't just pick up the jug. I'm a weed scientist and I was shocked. I was like, what in the heck? I mean, how did I miss this? So just keep that in mind. So you might say, well, this is a safer replacement to glyphosate, right? No, it is not. If you look at concentrated products that contain triclopyr, fluazepop, and diquat, they have way more warning language on those labels than glyphosate ever has had, okay? And the way they're managing that risk, that these are more toxic herbicides than glyphosate, is that homeowners will only be able to buy diluted formulations of this. So they're controlling, right? Risk is in the dose. They're controlling the potential dose that a homeowner could get. And then it's still going to have more language on the label warning you that there are verified risks with this herbicide. And so you can't have a frivolous lawsuit saying, well, if I'd known, I wouldn't have done it, because this label, they have proven the risks and they will state them on the label. Glyphosate didn't have those risks on the label because they were unfounded risks, right? EPA doesn't let you put non-confirmed risks on a pesticide label. So, okay, I don't know if I have any time left, but any questions, either general or specific, that I can help with? Hopefully gives you some things just to think about. Yeah. Think about I love the little plaque I have. 1 year's seeding equals 7 years weeding. But in some cases, it's worse than that, right? So you think about a pigweed or lamb's quarter seed. How long does it last in the soil? 50 years, up to 50 years. So every time you put weed seeds into that seed bank, you will manage them in the future. So I hope by fallow, as we look to some of the restrictions coming, that we're talking about managed fallow. If you let a piece go to weeds, then yeah, good luck getting it into production. Yeah, that's a great point. And years ago, we came up with a system in sugar beets that we called the micro rate. And it was way reduced rates, which probably in retrospect wasn't a wise thing, right? But it worked really well for some growers. And why was that? Because those growers had kept their beet and onion fields absolutely weed-free for years. So the weed pressure, because they'd never let anything go to seed, was very low. And so things would work on their farm that a grower who was battling, a standard weed pressure couldn't. So yeah, if you're faced with those, yeah, come up with something, mowing, cover crop, something that you can not just leave it bare, or yeah, it'll be a problem. Okay, I'll be around if you have questions, please. Oh yeah, Curtis. Oh, puncture vine. Yeah, that's a tough one. We worked a little bit with Cody Zeisiger on some trials, and I think we found that the Rimsel, the Matrix was one of the best ones, for that. But just timing's a killer, and then getting appropriate moisture and incorporation. Does timing have anything to do with the blossom of the puncture vine? In what setting? Are you talking about, like post-emergence control? With that being a true annual, I would say the earlier you can spray it, the better. And one of the challenges with puncture vine is where kochia, probably 90% of the kochia all germinates in a 3-week period, maybe. Puncture vine, as long as it's got resource, just germinates all, right? So, Roundup works great on it if you spray it every 3rd day, but you just turn around and it's bloomed and gone to seed. So I think wherever you can find some pre-emergence products that can help with that helps a little bit, but we just don't have anything that's super great on it, especially in our crops, right? And even on roadsides, people are struggling because you need something that's really persistent for the long term. If you don't have puncture vine on your farm, don't let it come in. But it's tough, right? It's coming on every wheel that turns. So anyway, thanks. Yeah, we can visit later as well. Okay, thank you very much.
So our next speaker is Prakriti Nepal. She is a master's student at the Plant Soils and Climate Department. She's actually one of the last students from Dr. Drost, my predecessor. So Prakriti is working on biostimulants. I had the chance to work with her last year while I, got into Utah. So Prakriti, I will.
Hello, everybody. I'm Prakriti. I'm a grad student at Utah State. And today I'll be talking about a study that we did at Utah State on assessing biostimulants for onion growth and production under stressful conditions. Onions are a cool season monocotyledonous crop. And when we look at the onion production in United States in 2023, we found that around 138,000 acres of dry onions were planted, out of which we harvested 133,000 acres of dry onions. In northern Utah, onions are commercially grown on 1,700 acres, making an average yield of around 67,000 to 84,000 pounds per acre. Here in this image, we can see the life cycle of an onion plant. This is for 1 year growth. We generally start seeding in March, and then we harvest around August to September. Onions are incredibly diverse, and this diversity is reflected in several key characteristics, such as they have different colors. We have red, yellow, and white onion types. Red and white onions are generally consumed raw, whereas yellow onions are more staple in cooking. And then we have different shapes of onions. We can find it flat, globe, grano, as well as torpedo. And when talking about flavors, it can be sweet or pungent. Sweet onions are generally consumed raw, whereas pungent onions are more staple in cooking. And they have different market uses. They can be used fresh, stored, as well as used after processing. And now let's focus on drought and its impact on the crop. When we talk about drought, it is important to know that around 95% of Utah's water supply is through mountain snowpack. And it is also important to know that drought can vary from year to year, which I'll be showing in my further slide. Drought has several effects on the plant, as it reduces the water availability. It can stunt the plant growth and inhibit the root proliferation, ultimately altering the plant water relation as well as the nutrient status. Drought is considered one of the major constraints to crop production, leading to reduced yield and decreased crop quality. That's why we have implemented drought in our study, as well as we have also used some biostimulants to check whether these biostimulants can work to work against the drought and help crop grow efficiently. Here is the US drought monitor for Utah. As we can see, in 2023, only a less part of Utah was severely affected with drought, whereas in 2024, we can see most of the areas have abnormally dry condition, as well as some regions with moderate drought conditions. Now, when we focus on onion root system and water needs, as we know that onions have a shallow root distribution where most of the roots are concentrated in the top 1.5 feet of the soil, where the depth of the root penetration into the soil is quite limited. That is why the irrigation water that is deeper than 2.5 feet is not quite accessible to the onion crop. And when we talk about water demand in an onion crop, it is highest during the ball formation stage. And this image here, we can see that it visually illustrates how shallow the onion root system is. Now, as we are talking about drought, now let's move to plant biostimulants. So we use plant biostimulants in our study. So these are natural substances or microorganisms that can improve the nutrient efficiency, stress tolerance, and crop quality independent of its nutrient content. Plant biostimulants have several benefits when applied in a crop. These can be applied in the soil, and they can also be applied in foliar applications. It can help in the efficient use of nutrient and water. It can also help to build tolerance against abiotic stresses, as I already mentioned about drought, and it can also help to improve the quality and shelf life of the crop, ultimately increasing or improving the total yield of the crop, and it will also help in increasing the photosynthetic activity. Now, when we talk about biostimulant, it is important to note that it is categorized into two main types. We have microbial biostimulants that are derived from beneficial microorganisms, and then we have non-microbial biostimulants. They are generally organic substances. In microbial biostimulants, we have plant growth promoting rhizobacteria. So what they do, they colonize the root system and help in the better nutrient absorption of the plant, whereas mycorrhizal fungi, they colonize the root system, increasing the root surface area, and they can increasing the root surface area for better absorption of the nutrients, which is not quite accessible to the onion crop usually. And after applying mycorrhizal fungi, it can help with increasing the surface area. Then moving towards the non-microbial biostimulants, we have humic substances and seaweed extract. So humic substances, when applied to the soil, it can improve the soil structure for better nutrient absorption. And on the other hand, we have seaweed extracts. So seaweed extracts are generally rich in hormones as well as other bioactive products, which can help in the overall quality of which can help to improve the overall quality of the crop. And these biostimulants can be applied in several crops. And in our study, we applied these biostimulants in the manufacturer recommended rate. And here in this slide, we have those 7 products that we used in our study. So in our study, we used 3 bacteria-based biostimulant. 2 of the products were mycorrhizae-based. 1 product was humic acid-based, and the last product was based on seaweed extract. So the 3 bacteria-based product, we have Tribus original. It had 3 bacterial species. Then we have Continuum. It had 4 bacterial species. And the Spectrum DS, which had the highest bacterial species, that was 20 bacterial species. When moving towards the mycorrhizae-based biostimulant, we had Myco Apply. It only had 4 endomycorrhizal species, whereas we have Mighty Mycorrhizae as well. It had 16 mycorrhizal species. It had endomycorrhizae and ectomycorrhizae as well. And then for the humic acid-based biostimulant, we used Humapro 16. And then for the seaweed extract product, we used Kelpak. It contained soluble potash. And all of these biostimulants in our study, we applied in manufacturer recommended rate. And before doing our field trials, we did some greenhouse study to assess these 7 biostimulants, so we can narrow down the biostimulants that we'll be able to use in the field. So the greenhouse study, we conducted the greenhouse study to assess the seedling emergence as well as the seedling growth after using all these 7 products. And what we found was Humapro 16, that was a humic acid-based biostimulant, and Spectrum, that was a bacteria-based biostimulant, showed improved seedling emergence as well as the seedling growth. That's why we selected those 2 biostimulants for our field studies in the year 2023 and 2024. So in our field studies, we used 4 onion cultivars. We used Merengue for the red ones, Walla Walla for the sweet, Bright White for yellow, and Vaquero for Bright White for white and Vaquero for yellow. And for the treatments, as I mentioned, we used those 2 biostimulants, Humapro 16, that was a humic acid-based biostimulant, and Spectrum DS, that was a bacteria-based biostimulant. Spectrum DS had 20 bacterial species. And for the Humapro 16, we did 2 applications, that was every 4 weeks. We did 1 application, we initiated in June, and then we did another application in July. And for the Spectrum DS, we just did 1 application that was done in June. And we also initiated drought stress as well as nitrogen stress in our study. For the drought stress, we used evapotranspiration data to estimate the irrigation run time. For the wet water treatment, we did full irrigation, that is 100% of ETR, whereas for the drought treatment, we reduced it to 75%. And I'll also like to mention that we only introduced drought during August. That's when during the bulbing in August, that's when onions need more water, and that's when we get water stress in Utah as well. And for the nitrogen, we used two different rates. One was recommended, that was 150 pounds per acre. And then we have low, that was reduced rate to 100 pounds per acre. So here I just have the yield by cultivars for the year 2024. So what we found when looking at 4 cultivars was Vaquero onion cultivars had significantly higher yield compared to the other cultivars. We also found that Walla Walla onions, the sweeter variety, had significantly lower yield compared to the other cultivars. So for here in these slides for the total yield and onion yield by market class, I'll only be showing Vaquero onion cultivars under 2 different water treatments. So what we found was higher irrigation treatment had improved total yield across all the treatments. Either it was control, Humapro 16 or Spectrum DS. We also found that Humapro 16 receiving full irrigation and Spectrum receiving 100 full irrigation had little to no colossal onions when compared to control or Humapro 16 receiving lower water or drought conditions. And most of our onions were jumbo sized, and only a small proportion of onions were either medium or smaller size. As we can also see here, full irrigation had higher yield compared to the drought treatment that was initiated in the month of August. Here in Humapro 16, we also saw higher total yield, as well as in Spectrum, we also saw higher total yield compared to the drought water treatment, as we can see in here. Now let's talk about nitrogen in onion production. Onions have specific nitrogen requirement requiring about 150 pounds to 200 pounds of nitrogen per growing season. Due to the shallow root system of an onion crop, it has poor nitrogen use efficiency. That is why it is recommended to have split application, which will minimize the loss and improve the uptake of the nitrogen. It is also so we should also not apply nitrogen late in the season or in excess amount, as it will lead to poor maturity or poor storage quality of the onion crop. Looking at the high fertilizer cost, it is an economic challenge for farmers to use higher rate of nitrogen. That's why we did this study to see if the reduced rate of nitrogen can actually improve the yield or improve the yield while using the biostimulants or not. So here's what we found in our study. So this is the yield for Vaquero onion cultivars under 2 nitrogen treatment and under 2 biostimulant treatment, Humapro 16, a humic acid-based biostimulant, and Spectrum, that was a bacteria-based biostimulant. So what we found that higher proportion of colossal onions were found when onions were treated with no biostimulants, but with higher rate of nitrogen, that is 150 pounds per acre. And we also found though they were treated with Spectrum, that was a bacteria-based biostimulant, it had a higher proportion of colossal onions. Even in this table, as well as of the water treatment as well, we saw higher proportion of onions were jumbo sized and lower proportion were medium sized or smaller size that is non-marketable. And we didn't see much of the total yield difference. However, we saw that applying nitrogen at higher rate had better yields compared to applying nitrogen at lower rate, even though we were applying these biostimulants in our study. And moving to on-farm trials. So for the year 2024, we did some on-farm trials with our farmers here. And this trial was done to assess the biostimulant effects on onion growth and yield under growers' conditions. So we had 2 locations that we did our study. One farm was at Ogden and another farm was at Bear River. So for our experimental design, we had 15 feet, we just chose 15 feet long plots, and there was 8 beds per farm. So in the farmer's trial, we used all 7 biostimulants, whereas in our study, we only used those 2 biostimulants that we selected from the greenhouse trials. So in here, while applying the treatment, we used a watering can, and then we calculated the biostimulant according to the manufacturer recommended rate. So for the yield of the farm 1, we found that when we applied no biostimulant, we applied seaweed, we applied mycorrhizae based biostimulant, the total yield was significantly total yield was lower when compared with the ones that was applied with Humapro 16 or bacteria-based biostimulant. We also saw that colossal onions were higher in the ones that were treated with Humapro 16 and Spectrum DS. I would also like to note that among these treatments, the seaweed was applied through foliar application and all the other treatments was applied in the soil. And the Humapro 16 was applied every 4 weeks, whereas Continuum or bacteria-based biostimulant and Tribus original was applied every week, whereas seaweed extract was applied every 2 weeks. So we did not see much of a difference in the yield among the other treatments, that is the bacteria-based biostimulant or the humic acid-based biostimulant. And moving to the yield of the farm 2, we found higher proportion of onions were jumbo-sized onions, and the total yield on this farm was pretty good when applied with these biostimulants, as it was also good in the control. The higher total yield was observed in the control rather than when we applied the biostimulants in these farm trials. To conclude, in our greenhouse study, we found that Humapro 16, that was a humic acid-based biostimulant, and Spectrum DS, that was a bacteria-based biostimulant, showed improved seedling emergence, leaf area, as well as biomass in greenhouse conditions. We observed no difference in the total yield of 4 onion cultivars when they were treated with biostimulants or a control under 2 different water treatments or 2 different nitrogen treatments recommended or reduced in the year 2024. And all the onion cultivars, they exhibited uniform responses under both drought as well as reduced nitrogen conditions in our field studies. However, there is a need for additional field trials, which is necessary to confirm the effects of plant biostimulants on onion growth and production under these adverse environmental conditions. I would like to thank all of my advisors, lab technicians, USDA Agricultural Marketing Service Specialty Crop Multi-State Program for this grant, Utah State University's Agricultural Experimental Station, and our Utah onion farmers for allowing us to do the on-farm trials, and all of the graduate student and undergraduate student that helped me during this research. And I'm offer any questions if you have. Thank you.
How did you guys select those properties, research-based information for biostimulants? You guys decide on.
So I think they wanted before. I think when they were writing the grant, they were trying to focus on ornamental plants as well. But then they switched to vegetable crop and watermelon. And when talking about the products, we selected the products that were readily available here. It was not based on prior research or anything like that. These products can be easily found here. Only the seaweed extract one, I think it's manufactured in Africa, but other than that, we can find it here readily. So that's why we chose those fast. Yes. Yes, except for the seaweed that was done through foliar application, all the other products were applied in soil. Increase.
Reducing the
No, I haven't seen any papers with the diseases and the biostimulants.
Our next speaker is at the IPM lab, actually. So he's gonna talk.
Okay. So you can see, oh, that's better. So you can see on the bulb mite, or here's like a onion where you can see a bunch of them just slowly crawling around just on the outside later. And again, remember their feeding can allow for just soft rotting and then the shriveling of the bulb. So here's a garlic clove where you can see that rot starting to take in. And here's an onion as well. Out in the field, you might notice stunted growth or just weaker plants. Early in the season, the bulb mites can cause delayed germination or poor seedling emergence. You might see brown corky scarring on the bulb. So here's a garlic clove where you can see these little brown spots. That's attributed to the bulb mite. And of course, like I've reiterated, this feeding is going to increase the susceptibility to different fungal and bacterial pathogens in the bulbs. So how can we manage bulb mites? So unfortunately here in Utah, we don't have an established threshold like we do with a lot of other insect pests and other crops. And I was looking around really other production areas in Washington or Georgia, they don't really have a threshold established either for bulb mites. So I think that's something for ongoing research. But as the growers, when you're out in the field, you definitely want to include bulb mites with your scouting. So looking for the plants with those signs and symptoms that we just discussed. When we're managing, we first and foremost just want to start with preventative measures. So obviously source clean seed and transplants with your onions. Clean the bulbs before storage and store at the appropriate temperature and humidity. In extreme cases, I've read of farms and other production regions where they will fallow the fields and just allow that organic matter just to decompose completely. Because remember, that's serving as the overwintering and food source for the bulb mites. There might be scenarios where you need to avoid planting successive crops of onions or crops. And then one area in Washington I read, they would do flood irrigation, or they would have periods of heavy rains during the winter, and that really had an impact on the mite levels that they found. So these are kind of all kind of more extreme measures, but I will show some different miticide options here at the end when we talk about the other pests. So let's switch gears and talk about beet leafhopper and beet curlytop virus. So I can't talk about this without mentioning Dr. Elmer Ball. He was an entomologist in the early 20th century at the Agricultural College of Utah, which is now here at USU. So he's the guy that really kind of pioneered the beet leafhopper and beet curlytop research. In 1917, he published this 56-page guidebook on some of the limited prior work and a lot of his own work as well. So this is actually still, I think, kind of a relevant guidebook. You can read it on the USU Digital Commons online. But essentially in the 1899 and 1900 is when we saw the first documentation of the beet curlytop virus. There was an outbreak in California's Central Valley in the sugar beet industry. They found this virus, and since then it's quickly spread to other Western states, including Utah. So it gets its name because obviously the first host crop they identified it on was sugar beets, so beet curlytop virus. However, today there's currently over 300 different host plants, including a lot of our vegetables and then different weed species that can harbor the beet curlytop virus. So the virus is exclusively spread by the beet leafhopper. So leafhoppers are really small wedge-shaped insects. The beet leafhopper is only about 3 millimeters long. It has this membranous wings that kind of fold over its body like a tent structure. The color variation can go from yellow to green to this tan brown color. So you can see, I put two pictures. So this is the same species, just different color variation. So that can vary by location, season to season. And unfortunately, a lot of other leafhoppers are also green and brown. So they look very similar. And it requires kind of a microscope and an entomologist to look at the key distinct features. So the nymphs, they're very similar in appearance. Obviously, they're a lot smaller and they have the undeveloped wings. So they're going to be a lot less mobile than these adults that move in the rapid short flight movements that kind of resemble hopping, which gives them their name, leafhopper. So the virus itself, the symptomology will vary from crop to crop, but I'll talk about here on tomatoes. So the virus is a curtovirus in the family Geminavertidae. So some of the main symptoms we'll see in tomatoes are these thick, crisp leaves that roll upward as the petioles of the leaves start to roll downward. So that's why we get that curly name from. The plant will become very discolored, almost kind of a yellowish color, and then some of the veins will look purplish. Infected plants will also prematurely have fruit ripening. And if you're looking in a large tomato production field, you'll usually see the infected plants scattered throughout sporadically. So that's another kind of indication that the virus is in the field. Unfortunately, once the plants are infected, they're not going to recover. So they can't get better once they have the virus. So you can usually assume that just based off the symptoms that a plant has the beet curly top virus, especially if you've checked all the boxes of the things, but we can never actually know for sure unless we do a virology test, like in a diagnostic lab. But so that may not be necessary, but you can always have it tested to be for sure. And just like any virus, whether it's in a human or another plant, the severity of the symptoms can vary between. So like if I get sick with the virus, I might have a stronger defense, so I may not present as many symptoms as like another person who's more susceptible, or they get really sick from the exact same virus. So we can see that same concept in plants, where some plants present a lot more sick than others. And interestingly, a lot of weed species can get the virus, but they will be unaffected. So you might have pigweed or kochia that has the beet curly top virus nearby, but you won't know because they're not expressing symptoms. So the big question is kind of, how does this virus spread and how can we understand it? So the beet leafhopper insect themselves are really adapted to high elevation, semi-arid conditions, and they'll overwinter on the limited vegetation in areas that have been uncultivated. So kind of based off that description, Utah and like a lot of our other intermountain states are really well suited for the beet leafhopper. Early in the spring, when the desirable weed hosts dry up, whether it's on the foothills or in rangelands, that beet leafhopper is going to move into the valleys or other production fields looking for alternative food sources. And they'll spread the virus by feeding on the plant, obtaining it, and then feeding on a different uninfected plant and then spreading it with their piercing, sucking mouth parts. And interestingly, tomatoes aren't a desired host plant for the beet leafhopper. So they actually don't like tomatoes. So what happens is they'll feed on that plant, realize it's not what they're interested in, and they'll move on to a different plant. But they were there long enough just for the virus to spread. And really understanding how the virus spread is very complex. This is actually kind of one of the ongoing questions of research that our plant pathologists and some of students are working on. But basically with transmission efficiency, that's kind of a measure of how well an insect can move the virus. And it's really dependent on how much of the virus is in the beet leafhopper insect themselves, which is also dependent on the number of virus in the infected plant that has fed on, and how many times it has fed on the non-infected plant since last feeding on the infected plant. So there's definitely a lot of layers and complexity to that, which I think makes this disease really hard to manage. So when you're sourcing tomatoes, you won't, there are currently no tomatoes that are bred to have resistance to beet curly top virus. I know when you buy tomatoes on the seed packet or catalogs, you'll see varieties that are listed to have some resistance to like the tomato spotted wilt virus or tomato mosaic virus, but we don't have that for the beet curly top virus. Our plant pathologist has done a lot of work using reflective mulch to control thrips and aphids, other common tomato pests, because that shiny reflectiveness deters them away from coming to the plant. So in theory, the theory is that kind of this reflective could also potentially work on the beet leafhopper, but this isn't really formally studied yet. But it's just kind of an idea out there. And then several years ago, when I first started, we did some work where we would use row covers over our tomato crops. I'll talk about more in a second. But understand a lot with like these kind of cultural controls. The management effectiveness is again really dependent on your location, like the climate, what weed hosts are there. Is the virus already there? The planting timing, the material application, and of course the proximity to like those overwintering sites for the beet leaf hopper. So another list of challenges to consider. So there are insecticide control, specifically targeting the beet leaf hopper, can have varying effects, but it is really limited to just due to the behavior and nature of the beet leaf hopper with their quick flight movements and ability to go long distances and leave an area. So I'll talk more about that in a second. I'll show you. So in 2019 and 2020, we did a very simple experiment with our USDA Extension IPM program, where we went to 5 different farm locations across northern Utah. And we set up the experiment where we had tomato rows that were exposed, and then we had tomato rows where we would cover with a row cover. And we used a low-grade spun-bound fabric just to cover the plants entirely throughout the season. And then, of course, throughout the season, we had these yellow sticky traps in the canopy of the plants where we would check throughout the season and do actual counts for the beet leaf hopper. So we would have to collect those traps and then obviously take it to a microscope, sort it out from all the other insects and other leaf hoppers and identify specifically the beet leaf hoppers. And then from this graph where we have the 2 seasons, looking at the number of beet leaf hoppers over the course of the year, you can see there's definitely a lot more beet leaf hoppers in 2019 compared to 2020, which just kind of shows like every season is going to be a little bit different. But the general trend we found is obvious, which is kind of obvious if we think about it, that row covers were effective in excluding the beet leaf hoppers from accessing the tomato plants. And then I didn't post these, but we also did yield data. And then the plants that were under production had a lot higher yield compared to those that weren't. But of course, the challenge with row covers, especially on a large commercial scale, is definitely there's a big cost to it, a lot of labor and a lot of time. So that's an economic trade-off that should be considered. Here in Utah, a lot of farms don't do this, and other tomato production regions, only farms that do it have large teams and the resources to use row covers. So I want to talk about pesticide options too. So in the back, we have the Utah Vegetable Production Guidebook that we're talking. So definitely make sure to grab one before you leave today and grab a second one for another friend that may not be here. But this is the guidebook that's put together by, it was put together by Dr. Dan Dross, and now Milena will inherit the project, and we have entomologists that work on it as well. But in this most recent version, we have different pesticide tables. So if you are interested in different products, you can go to the onion section, and we have the different products listed. We have the brand name of that product, mode of action, residual days, and then the different pests that it targets. So you might see products that you already use. So you can look to see, so like here's bulb mite, so you can check to see if it's listed. So how we compiled that is we use the TELUS agronomy label search. So this is also a resource that you can use. You probably already use it for when you get your products. But basically you look for the state, make sure that the product is registered in that state. You can look for a specific active ingredient. So I'm interested in tomatoes as my crop. And then you can look for a specific pest. So it won't specifically show beet leaf hopper, but if you just search leaf hopper in general, that'll come up, and then you can search. And then it'll bring up a whole bunch of options. So you can see if your local cooperative has these products, but I always think it's important to go a step further. So I can select an actual product. So this is Azagard. And then I can go to the actual product label. So this will bring up the label. Then I can just do a quick search of, oops. The one, make sure, okay, tomatoes is listed. That's in a greenhouse, but here it's listed for in the field as well. So this product's okay to use on tomatoes. And then I can verify leaf hoppers. So this product here, it just says grape leaf hopper, potato leaf hopper, and variegated leaf hopper. So you might have to further ask the sales representative or someone at the manufacturer if this product is okay for beet leaf hopper. So that's just kind of how you can do a quick search for different products, because like you can see, there's hundreds and hundreds. So in this guidebook, this is not an all-inclusive list. There's a lot more. So you always just want to go through this and just double check and make sure different chemical options are good for you. So you can also apply this to like the onion bulbs or onion mites for, the bulb mites for onion bulbs as well. So definitely grab one of these vegetable guidebooks. We also have it converted into a whole website. So this is the Utah Vegetable Production website at extension.usu.edu/vegetable guide. You can also get the virtual version of the guidebook as well. But this website has a lot of good information, so you can select the crop you want to learn about. We have the different pesticide product tables. You can read more about different pests, so like thrips. We have images showing, and then different diseases. So it's definitely a good resource to use. It's mobile friendly, so you can pull it up on your phone. And then of course, you can always reach out to myself if you have any other questions. So do we have time? Perfect. Yeah. Yep, so there's no pheromone or attractant on that. We just have it, this one's a little high up, but we would have it at the plant level. So we would have those traps equally represented in both the covered and uncovered. So now I think it was about every 2 weeks we would take those traps off, replace them, count. Yeah, definitely. So like in 2020, you can see early June, we had a lot of the beet leaf hoppers, but as the season went down, we would only find like maybe 1 or 2 mid-June, late June. Do we have any models out there for Yeah, I think as soon as you transplant, I think that's a good time to have it. Unfortunately, we don't really have any solid models. I think that's an exciting new area of research. I know Dr. Claudia Nischewitz has kind of off and on worked on curly top virus and leaf hoppers. So we could look at the years of the diagnostic plant lab data and correlate when the samples were submitted and see if there's kind of a pattern with climate and when those samples are first being submitted or when growers are noticing it. I think that would be really interesting. Yeah. Yeah. Yeah.
So it's a good opportunity. The color of the cover. So we are using the white cover but as shade cloths, we have
Yeah.
colors. Have you ever, have you find anything the effect of colors cloth on, or maybe some other?
No, I think that's definitely an interesting variable. We had our row covers obviously on throughout the summer, so it was really hot. So we picked the lowest spunbound grade. It was Agribon 15, but I think shade cloth could be a factor. I know shade cloth is pretty big, so I think the leaf hoppers could get through, but if there's like a thinner one. In more recent years, we've used insect netting, so that's more transparent. It's still kind of white, but it's more transparent than this. So there's different options, but I think with the potential research, the different materials would be interesting to compare. Yeah, I think would be worthwhile. Yeah. Definitely beets. I think everything in the beet family. I know a lot of the chard, Swiss chard can get it. A lot of the we saw during this kind of era, we would look at a lot of crops. So I think Swiss chard was one, spinach, and the symptoms were really, I would think, a lot more pronounced. So here's a photo of like a beet, so you can see where it's almost fully yellow and kind of more curled. So I think that's kind of more easy to identify than on the tomato plant. So anything in that family, I think it was a few years ago we tested pigweed, it was, that we suspected just because they were adjacent, and they expressed no symptoms, but there was a lot that we did confirm that had the virus. Yeah.
So go back in the onions, and you have that the reflection.
Oh, yeah, yeah.
That's just a
Where did I put it?
roll out on top. Does that inhibit growth, or what does that do?
So this is just a plastic mulch. So I think a lot of growers will just use like the black or white. Yeah, we did it. It's like kind of a silvery, kind of a reflective. We used it, I think it was peppers. We did our trial with peppers, and then in more recent years, she's did it with dahlias. And the primary pest that we were just looking for was thrips and aphids. So it hasn't formally been studied with the beet leaf hopper, but this is just kind of based off a theory that it's effective for these pests, that it might have some impact with the leaf hopper as well. I think so. I think she's doing it for a third year.
Guys, are willing also to conquer.
Yeah, definitely. We did this trial on all commercial grower farms back in those years. So.
I have one last question.
Yeah.
You talk about the checklist box, how to know if it's a vertical.
Yeah.
For example, last year, my second day here. So, where can we find this checklist box, or do we have it? Vertical or something else?
So the points I just shared were very kind of summarized, but our fact sheet might be a little dated and might need updating, but I think a lot of the symptoms are still very relevant. So here we have them all kind of spelled out, kind of what growers have found. So this was from, I know in 2008 down in Washington County, they did curly top trials. So this is where a lot of that information pulls from. So this fact sheet, I think, is a good starting point. or if you want to read more in depth about the symptoms and kind of the disease cycle with the beet leaf hopper.
To the vegetable program lab. I will put the link to the end of my presentation as well. I'm building this website as we go there and find resources. Also a link.
One more quick question, Dave.
Yeah.
So walking through the tomato patch, start seeing tomatoes, you know, like maybe not quite even to that extent.
Yeah. So we do offer the testing at the diagnostic lab. I think it's $30, which I know is kind of expensive. But even if you just have one plant that you're pretty confident you can get that tested and confirmed, you can know for sure. And then from that point, you can pull it out even just to prevent the leaf hopper. I guess I didn't even say that. Yeah, definitely pull the plant out, prevent the leaf hoppers from eating it and then spreading that virus. But continually monitoring and then maybe try to catch some of the early signs that the literature describes with the symptoms and removing it. Yeah. Yes.
Yeah.
Oh yeah, our lab site too. So
Or just call, you know.
yeah, so it looks like it's still $30. They do the DNA virus testing. Yeah, so you can also send a photo, that would be
Okay,
cheaper.
So do you guys have any other questions? Let's thank our speaker.
Make sure to grab the guidebook.
Yeah.
We brought a lot.
Okay, so let's continue with our next presentation. So it will be crop insurance with Barbie Hill. Barbie is with you. Let's make sure that it's it's on so the people online at the Zoom meeting can, yeah, just put everywhere.
Okay. Let's see. Okay, so like she said, my name is Bari Hill. I work for the Farm Service Agency, which is a department inside USDA. For vegetables, we do have NAP insurance. It's non-insured assistance program. It provides financial assistance to producers of non-insurable crops. So onions, they're insurable, so we can't insure them. But vegetables and fruits, we can insure. It is at the catastrophic level. So it's a 50% loss. Eligible producers are landowners, the operator of the farm, or if you crop share. Again, it is for all crops that are not privately insurable. The eligible cause of losses are natural disasters, damaging weather, such as drought, freeze, hail, excessive moisture, excessive wind, or adverse natural occurrences, flood, earthquakes. Excessive heat, plant disease, insect infestation. Now, insects on itself does not classify as a natural disaster. We have to prove that it was probably drought, heat, something tying it to that natural disaster in order for insects to be covered. The natural cause must occur during the coverage period and before or during harvesting. You can purchase SNAP. The deadline for fruits, fall planted, is December 1st. And then all your spring planted crops would be March 15th. You do have to file an acreage report with the Farm Service Agency. That for fruits is January 15th, and then your vegetables would be July 15th. And your insurance does not attach until 30 days after the crop has been reported with the FSA. So the sooner the better. There is a cost. So just for the basic catastrophic level, 50% and 55% of the price is $325 per crop. But you will not pay more than $825 per county or $1,950 per producer nationwide. We can waive that $325 or the buy-up premium by 50% for beginning farmers, limited resource farmers, or socially disadvantaged farmers and ranchers. As soon as you notice a loss or you're noticing there's damage to the crop, you have 15 days to let us know. If you are going to continue to and plan to harvest the crop, you will just then, at the end of harvest, bring in your production. But if you don't, it's a total failure. We can send a loss adjuster out and they will determine that loss. The production reporting date is the later of the acreage reporting date for the crop in the subsequent year or 60 days after the normal harvest date. So typically by the end of the year, we should be getting all of your harvest production information. And on that, if you do have NAP insurance, even the good years, like if you have a good year, it's really good to bring in your production and let us know those good years. Because if you continue to have a bad year every year, then your normal year is bad, right? So we want to prove that you have good years so that when you do have a bad year, we can get you paid more money off of that production. So we have a county yield, a T yield that we go off of for new producers that haven't harvested something before. If you have harvested crops before, you can bring in your previous 3 years. But if not, we'll just take 75% of that county yield for your production for the first few years. So here's a payment example. So Farmer Jim, he has 100 acres of pumpkins. He has an approved yield at 200 hundredweights per acre, and the price is $5 hundredweight. He just did the catastrophic level for the $325, which is 50% of your yield, 55% of your price. So his price would be $2.75 a hundredweight. So you take the 100 acres times the approved yield at 200, and you get 20,000 hundredweights expected yield production. Your disaster level is the 20,000 times the 50%, so 10,000 hundredweights. Hail hits on July 10th, and he notifies the FSA office in writing and takes the crop to harvest. His production equaled 5,000 hundredweights. So he takes the 10,000, the disaster level, and subtract the 5,000 as actual production, which is 5,000. And then you times that by the price at $275, and it's $13,750. And then he didn't have a premium because he didn't do the buy-up option. And then his service fee was $325. Here's the buy-up coverage. I think I failed to mention that. There is buy-ups, so on vegetables, fruits, there's different levels you can buy up. So it's 60, 65, 70, and 75, I believe, and 100% of the price. So there's just those different levels. So this time it's the same example, but he chose 60% of the yield, 100% of the price. So his expected production again was 20,000 hundredweights. His disaster level is the 20,000 times 60%. So that'd be 12,000 hundredweights. Again, he'll hit on July 10th, and he notifies FSA. Took it to harvest, 5,000 hundredweights again. His payment would be 12,000 hundredweights minus his production is 7,000. And then he gets 100% of the price, so $5. So his payment would be $35,000. He'd have a premium of 100 acres times the approved yield at 200 times the price times the coverage level is a $60,000 guarantee at 5.25%. His premium would be $3,150 approximately.
Harvey, how do you establish that price? Dollars.
It was an example. I don't.
Is that standard rule of thumb or?
I think it's market price. I'm not 100%, but
So I get your attention, one more question.
Mhm. Yes.
Generally, rule of thumb.
So to purchase the insurance is by March 15th. Crop reporting deadline is July 15th.
What do you see most people in vegetables are making?
We do have some vegetables here in Box Elder County that have NAP insurance. It's usually squash, pumpkins, mint. We have some mint, I believe. I don't think we have too many tomatoes. Tomatoes is an option. Mhm.
So if they can buy insurance through their local insurance, that's where they're gonna go.
Yes.
But if they're then
Yes, that's correct. Yes. Yeah, it's right on, I think it's 2871 Commerce Way. Yep. Each county has its own, Cache County has one, an office. Weber Davis Morgan's combined. So. So it's per unit. And so it's basically your whole farm. So if you have a field down here in Perry and you've got one in Willard, and Willard was really good, Perry wasn't, it kind of cancels out your loss because it combines the two. Anything else?
here in Utah, actually, producing biostimulants. We got lots of questions about biostimulants. Which one should I use? Should I not? I know we did some research. It was the first time then, and UPM was doing that. But we have lots of research to still. We have several products, out there. You guys don't know which one to pick. And maybe Michael you can ask any questions as you want, as he was also a researcher before, before the company. Anyways.
All right. Well, first off, my name is Michael Van Oosten. I'm the director of biostimulants for Rhodesian Life Sciences. We're a fairly large company, especially in the biostimulant space, but I'm R&D, so I'm not going to try to sell anybody anything today. But I would like to raise awareness about the fact that we are producing a lot of biostimulant products here in the state of Utah. And I think that's something we should be proud of. I'm a transplant. I was originally from Montana, did my undergrad at Colorado State, my master's and PhD at Purdue. But I was very happy to move back west. So Rhodesian Life Sciences has been around for about a decade and a half. We have facilities in Pasco, Washington, Cape Girardeau, Missouri, North Lima, Ohio, and South Salt Lake here in Utah. We are a biostimulant and nutrient use efficiency company. We sell fertilizer enhancers, stabilizers. We sell rhizobia as inoculum. We sell micronutrients. We're the #1 provider in the country of all the ferrous sulfate. If you have any sort of ferrous sulfate, your product probably came through our Cape Girardeau facility or North Lima. We sell a lot of different single ingredient biostimulants, and what we make here in Utah are multi-ingredient biostimulants. That means that it's not just one biostimulant, but 2 or 3. So we talk about this as the MAC technology. Originally, the MAC technology was developed by Cytozyme. A lot of you who might have deep memories may remember that Cytozyme was a Utah-based, founded and owned company for many years. And in 2001, Rhodesian acquired Cytozyme. And after that, I was brought on to kind of modernize the portfolio and do some research. And we're very heavily invested in research and being able to demonstrate data for our products. A lot of our sales are international. We have a very strong focus on the Midwest and row crops for soybeans and corn. But we also make a lot of products for specialty crops. And most of the products we make for specialty crops are made here in So a lot of the technology was originally developed in '75, and some of the formalized ways of putting all of these organic materials and mineral nutrients all together in a jug and making it stable so it's not goop what comes out the spout was developed about 20 years ago. And most of what we sell today are products like Crop Plus Organic. Some of you who might do some row crops might use Seed Plus Graphite, which is another organic product. And they have a lot of different applications. And they're essentially based on fermentation metabolites, number of biostimulants, and a good micronutrient package. So just so you may not be aware, we're based in South Salt Lake. We have a R&D facility that's 7,000 square feet. We have fermentation capacity, and we have a lot of competencies in our facility in R&D. And we do that to develop new products, test new types of biostimulants, and see if we want to use them in our products. I'm going to skip this. And essentially, in the end, what we make is brown goo. We also make some brown or black gray powders. But what we do is we develop biostimulants that are water soluble, miscible in water for ready dilution. They're compatible with other tank mixes. So if you're going to put a fungicide on or a pesticide on at certain points, we design for mixability. And then you may see that a lot of people are out there buying just seaweed, just Q-Mix or something like that. Our technology is based on taking multiple biostimulants, putting it together, and trying to give the most robust package. Because like you see from some of the single biostimulant studies, one biostimulant might help the this situation. One biostimulant might help in this situation. But once you put seeds in the ground and you have a growing season, you don't actually know what your stress is going to be. This is really where biostimulants help you overcome adversity. Biostimulants do confer a growth boost, but they're really good at abiotic stress mitigation. So that's what we're trying to design and develop biostimulants for that purpose. Ours have a heavy mineral micronutrient content, very strong in zinc, manganese, iron, and copper, as well as a bit of molybdenum to help you overcome that stress, and some cobalt in some of our products to act as cofactors for beneficial microorganisms in your rhizosphere. And we develop everything with a 3-year guaranteed shelf life. We're adding organic material to a jug that does have a half-life, regardless of what other people in the industry might say. And so this is our same slide. So we have a very diverse portfolio made across different facilities, sold in over 46 countries around the world. Some of the things that might be most relevant to you folks here in Utah is crop plus organic. We also have like stover, stubble, digester. And then we have a lot of small, very niche specialty crop things, cider red. A lot of people grow cherries, apples, very popular product in the Pacific Northwest. We have calcium-based products. We have fungicide-based products. The ones in the boxes down here at the bottom, just the ones made. But you can see that we do fertilizer, additives, nutrients, biostimulants, biologicals, and we're also in the animal and aquaculture business. We sell a lot of aquaculture products for shrimp farms around the world, and chicken and swine and dairy cattle feed supplements. So I just want to say two last things, and then I'm not going to delay anybody getting their coffee or their bathroom break. When we design biostimulants, we really have a global audience. Our customer base is all around the world. Now that might be a farm in Brazil that's 30,000 hectares growing soybeans, or it might be a farm in China where they'll deploy on a daily basis 300 drones to spray fertilizer over their crops. Or it might be somebody in India whose farm is exactly so they might be out there with a backpack sprayer. This might be aerial sprayed over a crop. We have a lot of that in South America. It might be fertigation. It might be in-furrow application or side dressing. It could be very, very small scale. We try to design products that can be applied in all of these conditions and try to have them as compatible as possible with a number of different tank mixes. And what we make here in Utah is what we call MAC technology. That's fermentation metabolites from lactobacillus, which has over 300 metabolites that are beneficial to the plant. We have the nutritional supplementation in the form of micronutrients, and these things work together to do seed treatment, soil health, foliar elicitors, and most important is managing abiotic stress. So see here are some of our signature flagships, Crop Plus. Let's see, I don't know who sells chemical in Utah, but Wilbur Ellis, Winfield are some of our biggest customers that distribute products. C Plus, other specialty ones with specific micronutrients that are soil amendments. Oil conditioner. All right, that's all I really want to go into, so I don't take up your break time. But we are making ag products here in Utah, and that's something to be proud of. And if anybody has any questions, please approach me. I don't normally deal with customers, but I can point you at a sales guy.
So we are going to the break right now. So we have half an hour, ish. We would like to thank Barry River Valley Cook and Corey Thompson for sponsoring the break. So thank you, Corey. And well, I hope you guys two little things. I think for most of you who came late. So we have a list of, information that we are asking you guys to fill up. So you could, have your phone number and email updated so you guys can be posted. We can post things to you guys. The second thing is a survey. So if you guys could help me to fill out that survey, it will really help me to get that into my formal needs assessment. I hope to see you all after the break, and that's all. Feel comfortable. We have bathrooms close to the main area, and if you guys have any questions to the speakers, feel free to reach out.
I do. I love with
We'll see, but I'm hoping they'll redo it. We did it like it is last night.
You don't sell anything. You just get it.
Oh,
Yeah.
no, I left at 6 o'clock.
I left at like 7.
I had extra time. It was, yeah.
Because your investment.
And there was some traffic around.
Yeah. Because I'm like good in the past.
Came through like the last day. That is.
I can't. If
I left my office.
Yeah.
I said I would.
That's what I started.
So I'll give you.
Yeah.
Now I can miss. Someone here.
But Mike Levitt was elected 3 times.
Yeah, we don't. Oh.
I wondered about that.
I haven't had any trouble.
One of them, one
It makes me work and then you put a barrier now.
Now I'm like,
Actually, I think. I don't think they have a chance to keep saying. Probably more work. I wouldn't watch that. The cat is sitting on his chest. Very good picture. Oh, wow. City council.
You would always get like.
He was trying to count. Kelly's one of the questions.
I didn't. Like, I was like my office.
They run that they were.
I did.
Yes, what I'm talking about. Yeah, my brother used to sell that. You tell me that.
Thanks for
Yeah, it's been on. Smack you. I haven't seen you.
Yeah, and he's got to see me twice this way.
I tried to make him. I did.
But I look at two of them. One goes out of the port now, the other one goes out of. They've got things big, like they got Little Mermaid. They got little Yeah, so all the inside rooms on the second floor. When I was looking, all the rooms available. Or like a porthole, and my kids, I think, yeah, that would. Yeah, we went on one. We had a lot of windows.
I've been on one trip.
I think so.
Food and entertainment.
Yeah, I'm trying to convince me. Yeah. I got six. I got one. Yeah, I've heard you guys are super Yeah, I got it too.
Because he didn't submit
I asked for me.
What was he gonna try? Those I don't mind.
No, we bring it in, it goes on that shelf.
Quality distribution. Quality distribution.
No, I like it right. Yeah. So,
Oh, he did that for me too. He refused to let you buy lunch.
no, I know, he bought me and mine didn't. Go to Shack now.
I like the heck out of it. He's probably my favorite guy to do this. Yeah. I've got everybody down. Yeah, he like inherited a farm.
Well, the one you talked about cats. Which one?
Takes a couple times now.
I'm still laughing at I tried all that. It didn't work. My dad has. I'm on a
And I wasn't sure. Still have hot. I was on one team. I've been on. That's what I told. I also have yeah, so so I was there too.
So attention, everybody. Let's continue with the agenda. Hello? Let's have a seat, please.
Touch the lights.
Go. All right, so we have three more talks. Hello?
Just keep on going.
Yeah, we have three more talks, and I will be giving the next talk for those who came a little bit late and are wondering who is this teeny tiny lady, talking here in front all the time. My name is Milena Oliveira. I am the new extension vegetable specialist from Utah State University. It's a great pleasure to me to be here, to meet you all. Thank you for coming. Thank you for your, effort you put into being here today. I would like to acknowledge Mike Grace for all the help he gave, he put into being, organizing this meeting together with me. Thanks, Mike. So I will be talking about vegetable grafting. All right. So the title of my presentation is Stronger Rootstocks, Better Crops, Optimizing Watermelon Production Through Grafting Technology. So if you guys have any questions, I will be very happy to answer, by the end of the talk. Or you can just reach out later. You have my email here. It's not up here on the screen for the Zoom. So it's just milena.oliveira@usu.edu. And let's talk a little bit about grafting on vegetables. So I think you guys are very, very familiar with grafting technology on, on fruit crops, but it can also be done in cactaceae, like in dragon fruit. I have lots of experience working on that during my PhD in Israel. And then we have the technology being applied in vegetable crops. For example, here in the US, measured grafted vegetable crops are tomatoes and watermelons, but we have a increased interest for tomatoes, for cucumbers. It depends on the region. It depends on, the area, the requirements of, resilient crops under harsh conditions. So the vegetable grafting is actually a simple technique as in fruits. So the scion is our high value commercial variety. So it's a selected variety that you guys might grow, might pick. And then, so they are selected based for the high yield and also high quality. So we get this scion and then we graft or we combine, we join into a rootstock. And this rootstock should be stronger, right? In terms of root growth and soil-borne disease resistance. So basically, we try to get the best out of the two worlds here. We try to get the best of a crop, a variety that, is selected for a certain area. And then we combine it with a stronger rootstock for several, not only for the soil-borne diseases, but for other traits. And why do we care about grafting, vegetable grafting? Why is that important? Why are we, talking a lot more about, vegetable grafting nowadays? So the process itself, the process of wanting, so to produce a physical hybrid, we call physical hybrid, a grafted plant. So you have to cut actually two plants. So this wanting makes what we call cross tolerance within the plant.
So once you get, a secondary stress, the plant is already stronger. So that's the main thing about a technique, about this technique actually. It makes the plant to be stronger to a secondary stress. And these stresses can be either drought stress or salinity, heat stress especially, and cold stress. So we get this plant, very strong plant, very vigorous plant, and we can get, we can extract actually the best of it, which is high yield and quality. So this is a couple of examples that we can find in the literature. If you want to have more information about it, just please reach out. So we found in the literature several examples or several topics being discussed and prove actually that grafted can increase, for example, resistance to soil pathogens like Fusarium, nematoids, and some other issues. It can be also tracked by race. So salinity issues, plants can also survive under those conditions. Heavy metals, increase water use efficiency, and of course, improving the yield and the quality of the plant. Sometimes farmers say, so I don't like to work with grafted plants because if I don't select the right side, the right rootstock, my plants will taste a little bit worse. So you are right, you need to select actually which rootstock you are using. You cannot just go around and play, I'm gonna pick this rootstock. No, you must have a understanding of which variety are you picking for both sides, all right? So in a few minutes, I'm gonna pass a table with several rootstocks for either cucurbits and also Solanasian. So, and I will give a source out of that, so you guys can go there and see what are the problems you guys are having, either biotic or abiotic. And you can look at the list of rootstocks. And so I will give you the list in a few minutes. So the dual benefit or the dual impact of grafting is actually a production tool. So grafting is a production tool and the source of as a production tool. So grafting can speed up or it can accelerate the delivery of several traits to the farmer. So for example, breeding and selection, we know it takes a lot of time. And if you do not, rely, well, you have to rely on variety trials, to select the best varieties to your area, right? But if you use grafted plants, you can have more than one trait, in one plant, in one physical hybrid, right? So it's actually decrease, the time and the effort to find that variety to copy with the stress you're having in your area. So as a business opportunity today, the grafting demand is increasing. So it's a great opportunity also for nurseries, where as the grafted demand increases, so they can also improve or have like a little bit of production with this area. And you can combine two different plants actually. So you have a tomato here. It's a true plant, so for those who don't know, I have experience with that from Israel. So I was there while there was a research going on this type of grafting. Crazy grafting plants, but yes, we can do it. A little bit about the history of grafting. So vegetable grafting is actually not a new technique. It was developed in Japan in the 1930s, and then it started to spread around in the 1960s. So even, with a slow process of spreading around the world, in the US, for example, there is only like 10 to 15 years. That's what I could find on research-based information on that for Europe and also to here. But thanks to a group of researchers, mostly on the east side of the country, grafting, grafting on vegetable grafting is being more and more common for several types of crops, tomatoes, bell peppers, watermelons, melons, specialty melons. So as you can see here, most of those spots are places or states that research or outreach trials have been done so far. But as you can see, there is in the Intermountain West, there is a limited information. So to the best of our knowledge, today actually, we are the first ones who are, starting to do some trials on that. So my goal is to try to select, some rootstocks that can be suitable for a broad variety, for a broad spectrum of problems, of challenges, right? So we are working on that. Here is an estimated use of grafting in all sectors. So today the use of grafting in greenhouse, it's very well established, and in the field is increasing. So we have, like when we talk about greenhouse for tomatoes, for example, we have more than 20 millions of plants being produced, right? And for field, we have the second one with more than 6 million of plants being produced. Watermelons is the second one. And so we have several countries who are, working on that. There are also several other countries like Netherlands who have, if I'm not wrong, 70%, 75% of their tomatoes are grafted. In Israel, my experience started there, so most of the vegetable crops cannot be done just by the variety itself. It has to be done grafted. Why? Israel, middle of the Negev, middle of the desert. So almost 90% of the country is desert. So we have problems with heat stress, we have problems with soil-borne disease, and we also have a lot of problems with salinity due to the irrigation, right? We don't have water there. We desalinate water and then use the water in our crops. So we basically grow summer crops during the winter, which is way better to, manage the heat within the greenhouse. And my experience there is that most of the crops, the vegetable crops, have to be grafted. Otherwise, we would not survive a secondary stress like the ones that they have it there through the whole year. Anyhow, sources. So talking about vegetable grafting sometimes is challenging. You don't know where to find information. So I am starting to, put my name out there, trying to get people to, get in touch with, see which rootstocks they have tried before, so we could try it here. Since we have challenging conditions such as drought, salinity, cold, we can try, I believe we can try to select those rootstocks, and it will be a good source for the farmers. So I heard last year, if I'm not wrong, a farmer lost 30% of the tomatoes to probably heat stress or some other diseases. So grafted and selection of rootstocks are a better source to keep, the plants through the whole summer. So here's the vegetable grafting website. You can go there, and if you go to sources, you can find actually tables like that. I will pass around if you wanna to have it, you can. So you have tables where you can find several rootstocks that, they were this group of people, especially from Ohio State University, they are, studying, and
they started to select already those rootstocks. And so tables like this, we are passing. So if you want, I can send you, these tables easily. Here we have an example for cubics, since the presentation is for watermelons, for example. So we have all these rootstock variety for, being selected. And then if you go to the right side, you can see which company you can find, and then which disease, is this rootstock resistant. For example, we have even, I think they do even in races, like for example, sorry, they have several races, so they also have some rootstock selected for that nowadays. We have also abiotic stresses on your right side hand corner there. So cold stress is, for example, this first rootstock resistant to cold stress. Yes. So yes, there are resources nowadays on different rootstocks. So you guys, could reach out later and discuss if you wanna, wanted to try some plants this summer. So I am partnering with a company called Traditional. So Traditional is actually an Israeli company. I found them here. So it was a company that I was working in Israel on my grafted plants. And I was very fortunate to find them here. So they are sending me some rootstocks to try here, as I'm going to show you some results today. If you guys are interested to try some watermelon plants, and I think I can get some tomatoes too, please reach out and let me know, and I can give you some plants so you can try this summer. All right? Anyhow, let's just continue. Why did I start with research on grafting since I'm new here? So before coming to UTA, I started, reaching out some county agents and even some farmers like Kelvin Johnson from Johnson's Farm in Ransom. So I was reaching these people to see what were the challenges, the real challenges here. Since my career is based on dryland agriculture, I know the challenges are very, I'm very familiar with heat stress, with drought, salinity, high temperature. But I want to know to what extent I could, be willing to find a strategy in green last year when I got here for my first summer. So since melons and onions, are the top vegetable crops here in Utah, so I decided to try to get some plants, get some rootstocks to work on watermelon. So that's what I did last year. Again, the company that I am partnering is one Traditional. So they are very, very good. They are there for more than 20 years, I would say. So they can advise you to which rootstock to pick and which variety. For example, if you have a variety that you like the most and you want to see if you have a rootstock that is compatible, with that, we can do that, okay? So you don't need to change varieties, if you don't need to necessarily change varieties, you need to find a better rootstock, a compatible rootstock that you want to grow with. All right, so I did a couple of small trials last year. I did greenhouse work, I did field trials, research, and I did a field validation in one of the growers that was willing to cooperate. I also did outreach trials, so I gave some plants to farmers just to see if they would survive. They did survive. I've got so salinity and fusarium issues place. I've got around 98% of survival. And then I also did I also gave some plants to another grower who has terrible problems with fusarium. The rootstocks that I work with, they survive there, and we've got very, very high levels of survival. Okay. and we are hoping to continue, to explore more traits this summer. So I'm going to talk a little bit, in deep about the trial number 3 and the trial number 4, a little bit only. So we did a research, a small little research field trial in Greenville's farm. So it's our research farm in Logan. So the rootstocks that I used were Omer, so a squash interspecific rootstock, which is a fusarium, is a rootstock fusarium resistant. And I also used the Carolina rootstock, which is a wild watermelon. So it can give resistance not only to fusarium, but also to nematodes. So if you have those problems, I think those 2 rootstocks would be suitable for your conditions. I also did self-grafting. So I, due to my research-based career, tried several things. And then I saw that self-grafting has sometimes the same benefits of some rootstocks, but we don't actually know if the rootstock, that we do self-grafting, it will be resistant to some other pests and diseases. We need to, do research for that. Self-grafting is basically you get the same variety and you do the wounding, cut, and then you graft again. So again, cross tolerance, it makes the plant become stronger once you, make the plant undergo this first stress of the wounding, okay? And then we compared with non-grafting. The varieties that I used were exclamation, fascination, and sentinel. So 2 triploids and 1 seeded. But today, due to the time, I'm going to be talking a little bit about, just exclamation and a little bit of fascination. So the research layout. So we use 3 varieties, 4 grafted combinations. In total, it was 12 combinations. We did use 100% of the evapotranspiration, and also we decreased a little bit, we decreased 25% to see if the plants would survive. We used the same amount of water and we used the same amount of fertilizer. We used a guard for each plant. So plant spacing. When we talk about grafted plants, especially watermelons, we have to take in mind that these plants are super vigorous. So they cannot be, basically planted with a regular row space like 3 feet, between plants. They need to be grown wider. So you need to go wider. One colleague mentioned we were talking about the row spacing. He believes that the plant can be grown 4 feet apart and 10 feet between rows, just because the vigor of the plant it's just great. So we did use 4 feet between plants, 6 feet apart, and my plot was 12 feet with three plants only per plot. This year we want to go a little bit high, with the number of plants per plot, so we could decrease it will be better for our statistics. So we did lots of things. We did plant height, we did all the morphology, and then we did all the measurements on the yield components. But let's talk about the average of total yield, pounds per acre. For these plants specifically, I saw that, let me explain a little bit about the graphic so you can see the combinations here. So exclamation on Homer, which is the squash rootstock, exclamation on Carolina, which is the wild watermelon, self-grafting, which is exclamation on exclamation, and non-grafting. So, and you can see the colors. So on average, we saw that there was a 174% increase of using the rootstock, the squash rootstock compared to non-grafted.
And then we saw that there was an increase of 64% and 51% for the Carolina rootstock and self-grafted. As I said, there are some benefits for this variety, for this cultivar, exclamation. For the other varieties that I worked with, the patterns were basically the same. There was no difference when between treatments, drought, and control when we decreased it 25% of water, which means it's great. We can use less water, the plant is very strong, it can survive. Especially here, as you can see, so the roots, I don't have the pointer here, but you can see the Carolina on drought, it actually increased the pounds per acre actually. So it means that Carolina is actually stronger under harsh conditions such as drought. So under drought, we've got also very high increases on yield, 145% for Homer, Carolina increased 131, and self-grafting 43% when we compare to non-grafted. So basically, the simple fact of grafting a plant, a variety itself, it gives like almost 50% in, like, it doesn't matter which condition did you guys run the trial, which I think is really great. So this trial, it was actually the first time we tried. We had several problems last summer. So, but yes, so the plants were really good. And if you are wondering about the taste, soluble sugars, there was no difference between the varieties or the rootstocks when we talk about the soluble sugar, the sugar content. They were very good, like really good. I gave fruits around. Folks were wondering which cultivar I was using, and then I could not have the chance to do the to analyze it in the lab, you know, just to get folks to rate those varieties, those rootstocks, but they were really great. So the validation I did a small validation in Johnson Farm. So here you can see. So this is actually, this is the root study, The root studies, I will show you the results for the validation later. So the root studies, we are actually starting doing that because I am very interested to work on the below ground. So I always believe that the below ground can give you, you know, lots of information about, you know, your challenges and how you could manage your crop better, just looking at the below ground. So we can see that Omer, the first rootstock grafted on fascination. So fascination here is the result for fascination in the top. They were really, really strong. And we got this, we could, you know, excavate just half of the plant. James did that. And we were very surprised of the thickness of the plant. This is a squash rootstock. So we are also working, you know, getting some data on the root studies to see, to get, you know, some correlation with the yield and also how could this plant survive under harsh conditions such as drought. Okay, so the validation I did on the farm, the plants were 3.5 feet apart. I was interested to, you know, just give the plants to the grower and see how it goes compared to our research. So surprisingly, the yield went way high compared to our research. And I think it has mostly, you know, many issues we had with the first year, you know, of trial, but also conditions. For example, we planted in the farm, I think, around 20 days after, so the degree days matters a lot when you talk about, you know, plants, grafted plants. So Jake, for example, he plants it early, he plants it in May. So I planted in the 14th of June. Remember, I think you guys were here, and for most of the growers in the upper part of the state, we've got cold nights on the June 17th. So it was actually 3 days after I transplanted my plants. The results were, they were really yellow. I don't have a picture right here, but they were really yellow in the next day, but I did not lose any plants, which means that these plants, the rootstocks, were also tolerant to the cold nights we've got, right? So, so here are the 12 combinations I use. We are, so we can go by varieties, so you have exclamation, fascination, and sentinel, the same materials I use. And this is what we can see. I don't have a pointer right now, but if you can go with me. So you have in blue over there, the average fresh weight, you can see that the first one is actually Omer, so the rootstock, the squash rootstock. And as you go down, you follow the Carolina and also the self-grafting and the non-grafted plants. So you can see that the grafted plants were very, superior compared to non-grafted plants. And that pattern goes from 5 to number 8 are fascination, and then from 9 to 12 are the plants for sentinel. Okay, so I was very fortunate to do this trial and to validate even with a little bit of, difference between degree days. Quality. None of the fruits were also impacted by the quality, so quality was good. But the fruits were, what I noticed is that the fruits, the firmness was a little bit higher compared to my fruits in the farm. So the degree days, our days are getting shorter. So probably we don't have, lots of photosynthesis putting into sugars. It goes more for the ferments, for getting the plants become stronger. But I did not get any hollow plants, hollow heart fruits, or most of the fruits they were good compared to, well, in both trials actually. So a little bit summary of my presentation. So grafting increased the number of fruits, so with fresh weight and fruit, fresh weight itself without affecting the quality of the food. So we have a lot of papers out there where they analyze, and some of them can even increase the quality of the fruits depending on the sign used, because the sign will give us the quality actually. The rootstocks will give us the strength to other traits. So decreasing 25% of irrigation in my trial did not affect the results for none of the varieties. So we are planning to go even, higher this year. We don't know if we have funds for that, but I hope we will get some, so we can do a broader study on that, and also get more different rootstocks to work with. So self-grafting showed promising patterns versus non-grafted plants. Research on the new watermelons, cultivars or rootstocks are needed for Utah. We don't have any selection so far. So rootstocks variety trial would be probably nice to look at some traits. Grafted studies also on tomatoes or peppers, some other crops would be important to run here, especially on the high tunnels. So there are lots of information nowadays on the high tunnels. The use of grafting plants on high tunnels, especially organic, because this is a sustainable practice. This is a sustainable tool. It can run to different cropping, different production systems. So for high tunnels, for example, there is the literature actually is saying that it would be show some promising results, since the small acreage farmers are trying to look for push the dollars per square feet. So those plants would be a good source for them. So this is the picture of grafted and non-grafted. I don't recall exactly. I have written, but I don't recall so far which I think it's fascination due to the shape of the fruit. So we have the non-grafted one compared to grafted. They go crazy. So even they express weeds. We notice in our trial that they can express weeds just by the treatment. Here are some pictures from our research last year. I would like to thank James for all the help. We've got also students to help like Evan and Carter, who really helped me a lot last year. It was first summer, so we did work a lot, and I was very fortunate to have those guys work with me. Again, so this presentation will be on the Vegetable Production Lab. Is a different website from the USU Extension Vegetable Production Lab. And if you have any questions, please reach out. If you want to try some rootstocks this summer, let me know. Thank you. Oh, questions?
What were the traits in the Ulmer that you charged for, or that you picked for the rootstock? Why did you choose Ulmer?
Why did I choose?
Yeah.
Well, it was actually,
HO.
so I contacted them, the Tri-History, and they suggested, okay? So Ulmer was selected for the fusarium, actually. Yeah, my main goal was the fusarium. Since, my previous needs assessment that I did with some farmers before coming here, they mentioned a lot of problems with fusarium. They have lots of problems with that. So that was one of my main goals, was to try to see if those rootstocks would survive. Yeah, mostly.
When do you graft the plants? You do it in the greenhouse or out in the field?
Yes, we do it in a greenhouse. There is a whole process, for doing that. There are several types of grafting techniques that we can do. So my program is planning to do some workshops probably in a few years. Well, next year, starting from next year. Yes, you need to match the diameter of the rootstock with the side, right? So this process, usually, the nurseries or the researcher like us, we play around for a while and then we find the best, days to pick both plants, and then we do the grafting. Usually cleft grafting is one of the most common grafting. So I have a lot of resources if you guys wanna, have a look, lots of extension pubs if you wanna look, into that. And Florida, I know Florida is very big on trying to adapt to small growers, trying to make like small healing chambers so the farmers could try to do their jobs. That's also my goal, okay? To try to provide a way that the farmers could try out easily. Like I have already experienced with that. Some folks reach out and say, oh, I tried and it did not work out. So problem with the technique, it's not, suitable for the type of crop you are trying. What else? Days, again, the diameter has to fit on the side that you are selecting, and the healing chamber. So healing chamber is always very problematic, but there are several, again, pubs, extension pubs, saying that we can do that. Yeah. Any more questions? Sure.
With the short growing season that we have in Utah, is there any chance that some of these rootstocks could help have an earlier fruiting?
Yes.
Have the fruit available in watermelon tomatoes?
Yes. So those rootstocks again were selected because I was mostly focused on the disease. But yes, there are, I think, did you get a table?
Yes.
So there are some rootstocks that are cold resistant or tolerant actually. So yes, it's possible to do that. And I am planning to, if I have the chance this summer, I will try some at the hydroponics also, to get it, get the season done. So I will probably, or if you guys want to try it, please let me know and I can probably, try to get you guys some plants this summer. We still have time, yeah, to order. Yeah. More questions? Okay, so let's move to our next talk, which will be Evan. Evan is a master's student at the Plant Science and Climate. He is also he is the latest student from, my predecessor, Dr. Drost. And I am very happy to, have worked with Evan for, the past summer. Okay.
Great. Thank you very much. Excited to have this opportunity to talk a little bit more. It looks like we were talking about more biostimulants, more water knowledge. So I'll get into that. But just a little bit about me. Like I said, I'm finishing up my master's up at USU. I'm at the end of my second year. I've worked with Dr. Drost for 3 years as an undergraduate and been interested in vegetables for quite some time. So yeah, hopefully some of these things will be interesting and useful. I know Capri's already talked about some of her biostimulant research with the onions. Hopefully we'll touch on some different portions and it will be interesting. So for background in this project, watermelon, we chose watermelon because it's an important world food crop. We've got 7 million acres grown worldwide. It's also a fairly large crop in the United States, with 142,000 acres, grown mostly in places like California, Texas, Florida, Georgia. And in Utah, while we're not the biggest vegetable growing state, it's a pretty substantial amount of our vegetable acreage with 570 acres, and that makes it the 4th largest vegetable crop after things like pumpkin, sweet corn, and of course onions. So, and then when you take related crops, so there's 570 acres of watermelon, there's next to 200 acres of melons in Utah, that's 200 acres of squash on top of that, and then about 1,000 acres of pumpkin. So all of those together, the related crops, which are cucurbits, that's about 35% of the acreage in Utah. So while we're talking about watermelon here, a lot of you, I think, grow pumpkins as well, maybe squash, cucumbers, and things like that. Some of these ideas are pretty applicable if they're fairly closely related and respond pretty similarly to these things. So, and the reason we're looking at some of this, the challenges that we're seeing is drought. So this is August of 2022. This is around the time that they were writing some of the grants for the funding that started this project. And you can see that very large portions of Utah are in exceptional or extreme drought. A lot of the watermelon production areas fall into these areas. We've got places like the Green River down here, down other areas kind of throughout the front where you got production. And luckily the water looks a bit better. We've had some good water years in the last couple of years, but I think it's the same good. We're either in a drought or preparing for drought. So it's good to think about these things. Long-term drought has a big effect on watermelon production. It's a fairly water-intensive crop, and you need it especially in the later season during flowering, fruiting, and sizing. And then in Utah, because we're mostly coming out of snow on the surface water, even in the decent water year, we can have some seasonal water deficits later in the season, which again is especially problematic for watermelon production. And lastly, when we talk about fertility, fertilizer costs have been going up recently as well. Makes it hard to have the nutrients we need. And on top of that, improper applications was the problems with water quality pollution and environmental degradation. So we hoped with this study to address some of these problems with water and with fertilizer by using biostimulants. So when we're dealing with drought, we have a couple of tools in our toolbox. One of those would be precision irrigation management. So we can see here some plastic mold being applied into a vegetable field. That helps reduce some of the evaporative loss of water, to has higher water use efficiency. You can also see the use of drip. In that case, we use that with onions in some instances and other vegetables. We can also use things like soil moisture sensors, so we have a better idea of how much water is actually out there, and use like evapotranspiration models for getting precision application, and all those things can help us to deal with the low amount of water we experience. After that, there's some interest in deficit irrigation. That would be applying irrigation that's less than the optimal amount. So let's say in a watermelon field, I had 20% less water over a season, but I only get a 5 or 10% reduction in yield. You know, that's a reduction in yield, but when it comes to water use, that's a positive. Then we have antibiotic stress tolerance, which is grafting is a really good example of ways that we can get some better stress tolerance, superior genetics. I mean, looking at the new things coming out and how much better they are dealing with things like heat, things like drought stress or cold. And then biostimulant applications, a lot of that is helping with abiotic stress, which would be, you know, low water circumstances, and that's what we're going to look at. This is that's the way that it helps with drought. And then we have fertility management. I just wanted to put this up here as just a reminder of general good practices for managing your fertility. Looking for the 4 R's, you want to make sure you use the right source. You want to make sure, you know, we're picking whether it be an organic or a mineral source, whether it's slow release or readily available, whether it's a nitrate type form of nitrogen or ammonia, and things like that. So it's good to look around and make sure you're using the right source, using good rates. Excessive fertilizer application can really be detrimental to plant growth. In watermelon, sometimes if you put too much on, you'll get a delay in some of your flowering and your fruit set. So more fertilizer is always better. So going for the right rate is very important. And then timing your applications. I have here on the left the standard growth curve for most of these crops, where you have growth on the y-axis of time. And we see that early on in the season, not as much need for fertilizer application, and later on as well as where the tapers off. So trying to target active growing times in your crop is very important for putting it out when the crop needs it. And then finally, the last place, making sure it's accessible in watermelon specifically. I mean, there's really big or pumpkin too. There's a lot of area in between rows, so trying to put it in the row where the plant roots are, where they can get to it is important. And then finally, things like biofertilizer, which you might have heard of. Those are things that fall into the category of biostimulants, and it's a possibility for offsetting some of your fertilizer application costs through those products. So there's a lot of different biostimulant products. It's a pretty broad umbrella. As we've seen, there's some biological things, there's some non-biologicals. We have humics, we have seaweeds, there's protein and amino acid things, as well as some nutrients that are not required by the plant, but can be helpful. So there's a lot of different products that fall into this, but in the end, we end up applying either to the above ground portion or we can apply it to the soil. And we're tending to see the benefits of higher nutrient efficiency, abiotic stress tolerance, and then an improvement in crop quality. So for watermelon in particular, sometimes that means better fruit firmness, sometimes higher sugar contents, better rind thickness, so the sugarability or storability is better, and then higher. So all those things can be really beneficial for production. So I'm just going to do a quick little overview of the two. I'm going to break this down into biologicals and non-biologicals, but plants have evolved, you know, in the soil system where there are living things going around them. And so it's interesting that we can sort of leverage these microbes to hopefully offset some of the inputs that we have in agriculture. Bacteria is one of those that's really interesting. They can do things like fix nitrogen. You can put some of them in there in studies that show they can get about 50 pounds per acre of nitrogen. It's just being put in there by bacteria. It's really interesting. You have availability of things like phosphorus, potassium, iron, whether that's through solubilization or chelation. And then you have these plant hormone production bacteria do that can change some of the architecture. They can stimulate some of that plant growth and also deal with stress response. So low water conditions that can be helpful for that. And then you have fungi. Fungi can help with acquisition, they can help with water acquisition, dealing with biotic stress and things like that. So very interesting products that I think we ought to keep an eye out for. Non-biologicals, we have things like humic substances, that would be humus, folic acids, humans, and a lot of those are soil applied and improve some of our soil characteristics, whether that's physical, chemical, wholly biological. But they also have some assistance in nutrient availability. They can help us with some hormone-like effect where they also interact with the root and stimulate growth in that sense. And finally, seaweed extracts, which help with nutrient uptake, stress tolerance, and plant growth. So while there are many more outside of these biostimulant products, these are the ones we looked at in our studies here. So that's why I don't want to detail on those. But there's a lot of things I think you've heard a lot of people use molasses and things like that. So a lot of those things fall into these. So we took these products. Here's a list of those. We've already seen them on the previous slide. But we took these in the greenhouse to see if they could help us with transplant production. A lot of watermelons are transplanted now, especially the seedless ones. They have a pretty hard time establishing the field. So high quality transplants is really important for watermelon production. And we wanted to see if these products would help us to increase the growth or increase the quality. So we have 3 of them that are bacterial here in blue. We have a humic acid product, a seaweed extract, and 2 ones. So in our greenhouse experiment, again, we were trying to find the efficacy of some products and growth. We selected, and then we wanted to select some superior products and take them out of the field after that. So we took 7 biostimulants applied with potting mix. So actually 6 of those were applied with potting mix, and the seaweed was applied later after it reached a certain stage as per the manufacturer's recommendations. Seaweed was grown for 30 days, and we took data on them every 10 days. I'm just going to show you essentially the final size of those, but we took measurements on the root, shoot biomass, we dried those the dry way to help split the leaf area. And in the end, we saw a lot of this spring trend, right? This is day 30 for the shoot fresh mass of a lot of these. And we see that there's a little bit of difference between some of the products. None of them are different from each other statistically, and none of them are different from control. And that tends to be what we saw through most of those metrics that we collected data on. But we did decide that there were ones that were top performers, even though it wasn't statistically different. So we selected 2 of these bacterial products and 1 of these mycorrhizal products and put those at the field for a field experiment. So like I said, we took those 3 products plus a control. We had 2 cultivars, so we used Crimson Sweet, Fascination, and then we imposed 2 different deficits. So we reduced water by 25%, but just in the seasonal. So it was about mid-July when we started to cut back on water. And then we reduced the fertilizer application by 33%. So we took all those, we had our own greenhouse grown transplants. We inoculated the media of those transplants just like we did in the studies in the greenhouse. We transplanted those and just used standard management. We had them on black plastic mulch, drip tape. We watered them according to evapotranspiration data that we were getting from the Utah Climate Center. And then we took data on survival, growth, quality. So, in all of the analyses that I've run so far, we haven't really seen much of a difference by the biostimulant treatments. I'm going to show you some of that data and go through some of the interesting things. I wanted to first start out. This is the half where we did reduction in irrigation, and so we'll look at the irrigation and fertilizer. And I wanted to look first out at the year. In the year 2023, at least up in Cache Valley, we had a really late winter. We had a really wet spring. We had a lot of rain also fall around the time of harvest. So the yields were lower in 2023 than 2024 by quite a bit, but that was just a year difference, and that's something we saw was different in some of the analysis. And then we also saw some differences in cultivars, so we can see that often, not always, right? So I've got here these Fascinating, Crimson Sweet perform pretty much the same, but in some cases, the Crimson Sweet and the Fascination were quite different from each other. So there was some difference in variety. That's to be expected. We know Crimson Sweet's an older variety. It's not a hybrid, and it performs differently than Fascination. But generally on here, we also see if we look at some of the biostimulants, again, not much difference, right? A lot of them are in the 70s, 80s, 80s. So we didn't see much of a difference in yield out of the field in the irrigation, but we also didn't see a huge hit in the reduction in water. I'll talk about that later, again, because it was such a high water year. We didn't necessarily see a deficit in 23. But even in 24, we had a decent deficit. We see maybe some reduction in some columns, but it's not consistent throughout. So then for the reduction in fertility, we see pretty similar trends. 2023, quite a bit lower yield than 24. Some differences in varieties. And we see a bit of a reduction in the yield with lower fertilizer, and that's kind of expected. But we wanted to see if the biostimulants would help with that low fertilizer. And again, we didn't end up seeing a whole lot of that. So that was all yield. This is sugar content. So we're looking at irrigation. 2023 to 24 was also a little different, but not by much. We see not much difference in the treatments, even though there's slight differences here and there. And then we see typically the fascinations were higher sugar content in some years, but not. So again, there's slight differences here and there. There's a lot of things, but there's not really a big through line with a lot of these. And the fertilizer kind of similarly, we have quite a bit higher sugar content in 24 rather than 23. But not a whole lot of difference by treatments, not a whole lot of difference by stimulant. So again, this is what we see, and I'll discuss some more of those in just a second. We also took these products out to some grower cooperative trials, 3 different places throughout the state, one in Cache Valley, one down in Spanish Fork, one in Green River. We applied all 7 of the products. We just took them into a hand sprayer, applied them to transplants. We reapplied it once a month and went out there and took some data. And I will just start by saying that, yeah, this is on-farm stuff. These were pretty far flung from where I was at, so they were being managed. I would show up and do what I could every time. And there were a few times where, yeah, by my mistake, I showed up after a harvest very happy, so that's why we can see these yields are pretty different. So in the control here, right, we have one farm, another farm, and then farm 3 is significantly lower. That's not an accurate reflection of the yield, that's just the data that I had, again, just because I had missed, they already harvested almost everything. So just putting that up there to show that there's some farm variability that's to be expected. We did not see any significant difference in these applications in the grower fields. And so that sort of, again, led us to believe that the things we saw in our replicated studies were pretty consistent. So for discussion, I would say that in 2023, our water deficit wasn't really a deficit. So we have to take that into account. They weren't different because they really got about the same amount of water. Even though we cut water, there was so much rain that it didn't matter. So also we use commercial products. A lot of the research on biostimulants is to do with like isolated species, often single species, and even single strains. And so it's a little different when you're using these commercial mixes. According to their doses, sometimes in the literature, with a lot higher application rates than we use. And then field conditions just makes everything difficult. Fields are very different, soils are very different, management is very different. So it's hard to always see in the field the effects that we see maybe in controlled environment, greenhouses, or in lab studies. So wanted to thank the grower cooperative. I got to learn a lot by going out there and seeing how different people grow watermelon. There's a lot of different ways to grow a crop. And that was interesting to me. And even though we maybe didn't get statistical results back that I would like, I got to see a lot of interesting things there. I just wanted to say as a conclusion that there was some variable effect of the biostimulants on the varieties. So leads me to think that maybe varieties respond differently to these products. That's something to keep in mind. Not every variety is going to behave the same with different products. So again, I think as we heard, targeting specific biostimulants for specific applications is important. And then we just saw that low fertility might have had a higher effect on one of these older varieties of sweet. So I think in the end, there's a lot of cool stuff going on with biostimulants. I think it's good to keep your ears around when it comes to those. I think we'll make a lot of strides forward in understanding how to apply them and how to use them. But again, in our studies, we didn't get to see any improvement yet. So I just put this up here. We brought a bunch of these vegetable production guides here, so grab one. And then I just wanted to point out, if you're interested in biostimulants, there's a lot of papers from different universities, just giving you a kind of a backstory of the 101, the down low, what they are, how you might use them, and hopefully we'll have them sometime soon. So with that, I just thank you. Thank you to the watermelon growers. Let us do some trials on their farms and all people help with this, and for the funding agencies, USDA. So with that, if there's any questions, feel free to ask.
Yeah. Question. So you applied soil microbes. Did you check the microbial population before and after, like pre-application and post-season?
So,
Did you change the population at all?
yeah, that's a good question. That's something I'm very interested in, but we did, this is a pretty applied study, and we didn't have funding to do a lot of microbe work, unfortunately. I have some soil samples where we looked at the fertility, so I have some NPK and stuff, breakdown on this, and there's some nutrients and stuff. And I have some soil samples that maybe I can send out, but again, it costs a lot to analyze microbes generally. No, it's I never maybe biomass and stuff like that.
I think you could just dump the soil and some dry off, shake it, take it back to the lab, back down and
Okay.
that's cheap.
Okay. I'll have to talk to you after, maybe, yeah.
Going to do that, especially because this year I'm going to move all my good stocks to.
You don't have to mess around. You don't have to mess around. Freezing anything.
Just a regular, the inspection. Yeah, sounds good. Do you guys have any other questions? So we will go to our last speaker, which is Michelle. Thank you for being, waiting. Thank you, everyone. Thank you so much. So please do not leave. We will have some time for seed givers after Michelle. Michelle is one of our, how do you call it, extension friend?
Yeah. Program coordinator.
Michelle is going to talk a little bit about you guys.
Getting touch screen.
Michelle is going to talk a little bit about the transitioning to organic program. So this program is being around already for so far 2 years, and many folks do not know, about it. So she's going to talk a little bit about programs. So if you guys have any questions for organic transition, we can help you.
Thanks, Milena. Yes, thank you. Thank you for inviting me to be here today. I'm kind of like she said, just trying to get this info in front of as many people as I can, because it's a program that's available to you all. So just trying to show up at everything and talk about it. I'll talk a little bit more specifically about the top project in a second, but first I just want to give us a little bit of background on organic. As we've seen today, organic can be used to talk about, chemistry and things associated with carbon or a type of material that comes from living matter. So what I'm talking about is organic agricultural production. So of course, organic as a word has all these different definitions, but when we talk about organic practices, I really like to think about it in kind of 3 ways. And they're all connected, one of which is this organic movement. So the organic movement really, obviously organic production practices have been used for thousands of years. It was kind of the default prior to industrialization, but what we're talking about now is kind of this more modern organic agriculture movement, which really came about around the time that, post-World War II, there was a lot more development of these synthetic applications, synthetic inputs to put on to use in your operations. And there was a group of producers who were kind of saying, hey, we'd like to continue using practices that maybe align with nature, that lean less on inputs from outside of the farm. And we want to use these practices to farm. And then you also had a lot of consumers who were saying, hey, we are interested in purchasing products that have been grown using these types of practices. And so really what we were seeing here, and that was really around the time of kind of the environmental movement, was when there was really a consumer movement to say like, hey, this is something that we want to be able to support and buy. And so this is what I see as kind of the organic movement, was really these producers who were growing things in a certain way, and these consumers who were saying, hey, we'd like to buy things So how do we connect the two, and how do we get some sort of integrity within that organic system? And that was really what kind of was taking place over the 70s, 80s, and 90s. Was there were more certifying bodies saying, hey, this is how we're defining organic. There were different groups saying this is how it should be defined, this is how it should be enforced. And this is where we really see the birth of the organic regulation. And it's kind of funny because it really was pushed by producers saying like, hey, we need some sort of standard here. We need some ways so that we can say, you know, I'm Joe at the farmer's market, I'm growing organically. And that, you know, Betty down there says she's growing organically, but we use totally different practices. How do we make this kind of more cohesive? And so we really saw a push for organic regulations to come about. And it wasn't until 1990 that the Organic Food Production Act was passed. And so this was kind of the first law. So they really did turn to the federal government to decide that this is how we're going to define organic. And so it was in 1990 that the Organic Food Production Act was passed. And then it took 10 years of debating to really come up with how are we defining this, how are we enforcing it, how are we administering it. So in 2001, the National Organic Program was created. I'd like to give this much background just because I think there's a lot of people know the word organic, they see the word organic around, there's just some confusion about what it means, what it doesn't mean. I'd just like to give people a little bit of background. So then if we move on to today, the organic industry. So organic really has become an industry. In 2023, there was $69.7 billion in organic retail sales, which to me seems really big, but I think it's like 5% of total retail sales, so it's still pretty small. But it has been consistently growing since 2000, since 1990. It has been a consistently growing market. It spans local producers to global commodity markets. We're talking about, you know, people at the farmers market, but we're also talking about, you know, global commodity markets. So I just like to emphasize that it isn't just kind of these small scale producers. It is also mid-scale, large-scale, international industry. So kind of this three-part but related movement, regulation, and industry. So how is it actually set up today? So the USDA National Organic Program, so the National Organic Program, NOP, is the rule. That is like the written regulations of what it means to be certified organic in the US. So the USDA, using the National Organic Program, accredits certifying bodies. So there are public and private certifying bodies. So you could, most state agricultural departments have an organic program, like USDA has an organic program. There's also private certifying bodies. For example, Oregon Till, California Certified Organic Farms. Some of these might sound kind of familiar. So the USDA accredits those certifying bodies, and the producers can choose kind of whichever certifying body they prefer to get certified organic. There's technically 4 scopes, but livestock, processing or handling, and then crops. The 4th one is wild crops. I think it's technically a separate scope. So if you're a producer, so a handler would be somebody who processes organic materials and sells them as organic. So like a bar or a, I don't know, a shake or something like that. So all of these folks, and I like to emphasize this because every organic product that you purchase in the US is certified to the same set of standards. And I think there's sometimes some confusion because that accredited certifier label will be on there, so it seems like, oh, they're kind of accredited, certified by all these different people, but it's all to the exact same set of standards. And that was really what that development of the regulation was doing, was making sure that there's one standard that everybody's being measured against. So what is it? I really like this OATS definition because I feel like it kind of encapsulates all 3 of the things I pointed out on the last slide. So it's a set of production practices and principles rooted in natural systems. It's defined, regulated, and protected by the USDA. It provides a verification method for consumers and a market premium for farmers. So we're really getting in there. It's a production, it's kind of a philosophy, it's a production practice. It's defined, it's regulated by the USDA, and then these benefits are that it's kind of communicating, like I said, between consumers and producers, communicating those values and practices. So what are some of the things that those that the National Organic Program rules actually say? So organic production, organic operations must utilize crop rotation. It is written into the regulation that it must maintain or improve soil, water, and biodiversity. Use preventative weed, pest, and disease control first. So kind of like we've been talking about today, or with the weed management, kind of that IPM thinking where you're like, first and foremost, we're using cultural practices, we're using prevention, we're using all of these other things, and then only down the road, if necessary, are we using some sort of input. You're keeping verifiable records. There is a lot of paperwork involved in organic. So I don't like to sugarcoat that part of it for sure, but we're keeping verifiable records, and that's because we're really trying to maintain that integrity. No use of prohibited materials. I realize that is super big, but you can go into the National Organic Program regulation, and there are two main lists, and these lists include materials, not products. So it's not going to be a list of like products you can and can't use. It's going to be a list of actual material in that product. And one of the lists is there. It's a little bit funny how they do it, but it's one list. So it is assumed that all naturals are allowed unless they're on this list of prohibited naturals. And then the second list is it's assumed that all synthetics are prohibited unless they're allowed on this list. And so it's two different lists that are just kind of, you know, it's obviously very hard to be all inclusive and name every single natural substance that might be used in production. And that natural versus synthetic is pretty well defined by the organic regulation. And there are also, you know, the OMRI is a label you guys might be familiar with, and that's the Organic Materials Review Institute. So you might see that on a bag on some input that you would bring onto your farm, and that is a whole organization that is essentially a new product will come online. They'll apply for OMRI, and OMRI will look at the ingredients, look at, you know, both the active and they'll look at all the ingredients and make sure that it's either allowed or not allowed in organic production. Washington State Department of Ag also does this. A number of places do that, and so it doesn't have to have just the OMRI label, but just FYI, that's how they're kind of reviewing those materials. And then use of conservation tillage practices. So like I said, it does have this imperative in the regulation that you are maintaining or improving soil health. And so it is asking that you are using conservation tillage. It's not no-till, but you still need to be maintaining soil health. So benefits, we've kind of touched on some of this. Producers receive a market premium for their products. Obviously, this varies a lot, what product, what market, but in general, that is a trend that we have seen. Consumers can trust that their food was grown using organic principles. This is important to people, and it is important to people in Utah. I think I get a lot of feedback that like Utah doesn't care. And it does, you know, not the whole market. There's definitely less interest than in some other states, but there certainly is a market there. There is a niche market there. Environment is benefited through soil, water, and biodiversity conservation. So it's kind of this three-pronged benefit that we're seeing. And then just some of the benefits to other benefits, farming without synthetic fertilizers and pesticides, and, you know, maybe reducing some of that weed resistance that we talked about earlier today. But you are also, yeah, some people just that I talk with just say, yeah, I like to not use that stuff. So that's great. You're also maybe reducing some of your input costs. Farming practices that protect natural resources and biodiversity. So you're really leaning on beneficials, having, you know, pollinator patches, stuff like that. Organic is non-GMO. So a lot of people are looking for those types of labels. Organic always means non-GMO. You're, like I said, attracting that kind of niche consumer base who value those same practices, so you're able to kind of meet them where they're at. And in that same vein, it's a tool for local producers to increase revenue and expand market opportunities.
if you're small and you want to get into a grocery store, this could differentiate you. If you're at a farmer's market and everybody's selling the same thing as you are, this could differentiate you. I really do think that it's a way for you to kind of tie your story. If these practices do align with your story, then it's a way to kind of communicate that to consumers. And you can use the word organic if you're certified organic. For better or for worse, it's technically illegal to market your materials, your products as organic if they are not certified organic by the USDA. So just FYI. But if you get certified organic, then you can use these claims and you can use the USDA logo on your products. So how do you get certified organic? Also, please stop me at any point if anybody has questions. I don't mind doing them while we go. So the biggest thing that's going to matter in how you get certified organic is if you're currently using organic practices. If you are not, AKA if you are using prohibited materials now, then your land needs to undergo a 36-month transition period. So 3 years. If you're not using prohibited materials, you can be certified or you can sell your product as organic 36 months after the last prohibited material application date. So if it was 4 years ago, then you're good to go. If it was 1 year ago, you've got a little bit to wait. So first and foremost, figuring out what you're doing right now that might not be allowed. Learn what it means to be organic and if it's right for your operation. It might not be right for every operation, and that's totally cool. But if it does seem like it might be a good option for your business to explore, or maybe you're already using organic practices and you just want to get the certification, then you're already checking off step 1 here because you're learning about what it means to be organic. Choose your accredited certifying body and work with them to understand the application and fees. Each certifying body is slightly different. Like I said, they are all being accredited to the same standards, so their application should be asking for similar things, but they will have slightly different application processes and fees. You would create and implement your organic system plan. This is going to be the bulk of your application. It's essentially a plan that is, usually there's a template from your certifying body, and it just is kind of going through and asking, how are you meeting those regulations? And so you're kind of saying, this is where I get my seed. This is how I plant it, where I plant it, what I put on it, how I harvest it. So you're just kind of laying out, hey, this is how my operation meets these regulations. Then you're going to meet with you're going to send in that organic system plan. That's going to be your application, the bulk of your application. There'll be some other stuff. And then your certifying body will review it. If it seems like you are potentially able to be certified organic, they will send out an inspector. The inspector will actually come to your farm and they'll kind of just be the eyes of that organic system plan, and they're looking around to make sure that what you said you were doing on your organic system plan is what you're actually doing on your farm. And then they will give their report back to the certifying body, and the certifying body will make a certification decision. And then you'll be certified organic. And after that, you will need to update your organic system plan and be inspected yearly. This is not easy. It's not easy, and part of this program is to be sure that we are helping people if they want to go through this process. We're helping them get there. So maybe I'll go over this in a second, but I can help with, developing your organic system plan. I can talk you through some of the regulations. I can talk you through what an application might look like. So we are definitely here to help, but I like to emphasize this because it just is, maintaining integrity in that system. I mean, if you think about it, every organic product that you buy or eat has that operation has somebody coming to it and inspecting it every single year. So I do think that it is a little bit of a testament to, there's a lot of labels out there. This one is doing something, I think there's sometimes like, organic is BS. And it's kind of like, well, maybe it's not doing everything, but it is doing something. And we are, checking up on these farms every year. So just a little plug there. So what is the Transition to Organic Partnership Program? So this project, it's a national project. So every state actually has I have a counterpart in every state, but I actually work for USU Extension. So USU Extension is leading the program for the state of Utah, but we are part of this broader top network. Primarily what we're doing is just trying to increase the number and network of organic producers in the state of Utah and around the US. We do this through technical support, mentorship, community building, and workforce development. So really, it's just we're trying to make sure that there's a team there, making sure that you have all the technical resources that you need and that we're making them specific for Utah. Making sure that if you want to have a farmer-to-farmer mentorship, then we have that available for you. Workforce development, we're trying to make sure that there is a track at USU for folks that want to specifically learn about organic production, that they'll have that option at USU. And community building, stuff like this, just making sure that y'all know this exists. You guys know that NRCS exists. You know that UDAF exists, just like that everybody is talking to one another so that if this is a good option for your business, then you know where to go. Nice. So just one more plug for our mentorship program. So we're actively recruiting mentors and mentees. The mentors I kind of know because the mentors are already certified organic. And the mentees are folks that are interested in transitioning to organic. And so what we actually do is we match a mentor and a mentee for a year-long mentorship program. That might sound a little intimidating, but really it's about 40 hours over the course of a year of direct one-on-one farmer-to-farmer advisement. It can be more, but we're just trying to make sure that as part of this team, you got USU Extension, you have me, and then this is just like a farmer that you could actually call. And we try to match you with somebody that is growing the same thing as you on the same, on the same scale as you. So when you call, they can help say, oh yeah, this is how I solved that problem, or this is how I manage that pest. So there is a $500 travel and education stipend. That's just to help. We just want to make sure that this program is not burdensome. We're not trying to just give you another thing to do. We really want you to be excited to maybe go visit your mentor, or go to an educational event together. I think that's all I have. I have my little thank you here, but I'll go back to this one just in case anybody wants. That's our just application, or maybe it's better. Let me know if you need the QR code. Thank you. This is my contact info. I also have brochures about it. I have a lot of little marketing materials. If anybody's interested, I can definitely give you more info and give you my contact info. Any questions? I know I'm the last one of the day, so if you guys just want to leave, then. Cool. Great.
So it's actually a great opportunity because it's being recorded. So other folks, we don't even know if it's, well, in our county here or somewhere, if they wanted to have some information, they can, reach out. Okay, so we have a little bit of time. I'm sorry, but we have some seed dealers here, so some companies. If you guys wanna, come here right now and just talk about a little bit, we have like 2 minutes per person. So you guys wanna have it, just come here. Okay, sounds good. Let me just give you the mic. So no, the thing, it's for the record. Thank you for coming. Anyone else? All right, so I would like to thank you all for being here, for sticking with us until one, like 5 minutes only. I would like to, thank again you guys for being here today. We actually don't know exactly how are we gonna do this Northern Utah Commercial Vegetable Grower Meeting. So you guys have to tell us, okay? So in a couple of days or maybe this week, we are gonna have a evaluation for this meeting. So I would appreciate if you guys could, answer the evaluation for the speakers and also for the whole event. So things that you like, things you don't like. When do you, want to have this meeting? If you want to have it every year, every 2 years, if you want to have it online, if it's, easy for you to just come here. So just let us know, what you guys are, thinking about having this event from now on. I would like again to thank the speakers, thank all the growers who came here, and thank you so much, Mike Pace, for organizing this meeting together with me. So thank you guys and have a great day. Oh yeah, so the the pesticide credit. Yeah, don't. Yes, and please, if you did not update your email and your phone number, please do that. And we have the needs assessment, the small survey. If you did not have the chance to fill that out, I can send you by email too. Okay? So thank you guys for coming again and have a great day.